And we start with the first team, with the first two of brilliant minds here in the room. It's Patrick Cramer, one of the winners, and it's Christian Spahn. So their topic is breaking the wall to replicating SARS-CoV-2 polymerase. And I just have to get my slides. As all the affiliations are filling a lot of paper and all the awards you've been awarded and everything, so I decided to write that down and read it. So Professor Patrick Cramer is a chemist focusing on structural biology, and after his doctorate he joined Roger Kornberg as a postdoc at Stanford University, and Kornberg later won the Nobel Prize in 2006 for his studies of the molecular basis of eukaryotic transcription. Patrick played an important role in the lab in studying the RNA polymerase, which he says is in the center of life. So since 2014, he's director at the Max Planck Institute for Biophysical Chemistry in Göttingen and uses a technique called Cryo-Electron Microscopy. And when the corona pandemic evolved, he started research on the polymerase of SARS-CoV-2 and found out more about its structure, which makes us understand which drugs could fight the virus. So, warm welcome to Patrick. The matching talk partner is Professor Christian Spahn, and he calls another Nobel Prize winner his mentor, Joachim Schwanger. Frank won the 2017 Nobel Prize in Chemistry for his role in developing a way to create high-resolution 3D images of biomolecules such as proteins and ribosomes. And in fact, he is one of the inventors of cryo-electron microscopy, the technique Patrick Cramer uses, as well as Frank's mentee, Christian Spahn. He is the director of the Institute of Medical Physics and Biophysics here at the Charité, and will lead us through the years of discovery of the game-changing technique. So welcome. And as they are the brave two who start the day, so this is how we do it. Patrick will start with his 15-minute talk. Then I'll hand over to Christian. He presents his slides, and Patrick sits down again. And after that, after Christian has presented his slides, the three of us come on stage and discuss. So thank you very much.
Well, thank you very much for having me. Thanks for the introduction, and I would like to entertain you with our latest research on the coronavirus SARS-CoV-2 polymerase, and especially would like to emphasize how our recent work can be used to understand how antivirals that are actually used for treating patients, how they work, and also how it can help to develop new ones. Can we have the first slide, please? Thank you very much. So why do we need antivirals? We have great vaccines that are very efficient and very safe. I think we need such antiviral drugs because we need to treat people who are not vaccinated. There will always be some. Also high-risk patients, we need to prevent severe disease. We need to reduce the number of hospitalizations. Also, prophylactic treatment would be fantastic, for example, within families, within school classes, and so forth. And to control local outbreaks, even once the numbers will be down due to vaccination, once we overcome this, there will be local outbreaks. And don't forget, it's only a matter of time for new coronaviruses to come, and then we need drugs to treat these outbreaks before we have new vaccines. So there's actually three main targets in the coronavirus that can be targeted for development of drugs. You can either try and block virus entry. This is what nanobodies and antibodies do. You mask the virus so that it cannot enter the cell. You can also block protein synthesis, especially an enzyme called the protease. And last week we heard about the news from Pfizer with Paxlovid. That's a new inhibitor that can block that enzyme that is needed for the virus to make the proteins. And then finally, the third major target is the RNA polymerase, which is the central enzyme that replicates the RNA genome. And remdesivir and molnupiravir are the drugs that target this step of the virus life cycle. Don't read those formulas. This is just to impress you and tell you that many antiviral substances are actually inhibitors of such polymerases that the virus uses. You all know, for example, acyclovir that blocks the polymerase of the herpes virus, fantastic drug, but also it's used to treat HIV patients and other diseases. When the pandemic hit us, we were trying to make the enzyme, the coronavirus polymerase, in recombinant form and in functional form, and that's always the first thing you need to do. And we achieved that and could then use cryo-electron microscopy to solve the three-dimensional molecular structure of this enzyme of the coronavirus polymerase that you see here. This enzyme consists of 3 different subunits, and they have various roles. The key subunit, NSP12, is actually the catalytic subunit, and the other subunits, NSP7 and 8, are accessory subunits. They, for example, ensure that the polymerase, when it replicates this very long RNA genome, that it doesn't fall off the RNA. Coronaviruses actually have the longest known RNA genome, about 30,000 nucleotides. So we made the first movie of coronavirus replication. You can see it's a very simple movie. You can see the nucleoside triphosphate substrates flying in here from the right and the nucleotides being added at the active site of the enzyme to the growing RNA chain. And this is how copies of the coronavirus genome are prepared in cells that are infected with the coronavirus. So now you can use that polymerase enzyme as a drug target, right? If you would stop it, then you would stop the replication of the virus in cells. And this is what has been tried to be done with antivirals such as remdesivir. The problem is that the usual RNA polymerase inhibitors wouldn't work in the coronavirus. This has to do with the special function of the viral polymerase, a so-called proofreading function. But it was found that remdesivir works to some extent, and it actually causes an elongation barrier. So the polymerase is not stopped entirely, but there's a barrier to elongation. It's slowed down, and we could actually take several structural snapshots of the polymerase to make a movie of how remdesivir would interfere with viral replication. So here, this remdesivir active metabolite has been incorporated into the growing RNA chain, and then the polymerase does not stop, it rather incorporates 3 more nucleotides, and then, only then, you hit this elongation barrier where these atoms in the remdesivir moiety will clash with atoms in the coronavirus polymerase. And this is not, a total inhibition of the enzyme. It's only a barrier. So when there's high concentrations of nucleoside triphosphates, and also when you wait for some time, then this barrier will be overcome. And that is probably part of the reason why remdesivir is not such a successful drug. Also, when it's given late, it has to be given by transfusion to patients in the clinic. When it's given late, replication is already through and the patients suffer from, diseases that are caused originally by the virus but are not due to the actual replication of the virus. So remdesivir didn't bring a major breakthrough in the clinic. And then, companies went to another one, molnupiravir by Merck. And we were also establishing the molecular mechanism of how molnupiravir would work. And so what we found is that the active metabolite of this drug is able to be incorporated by the SARS-CoV-2 polymerase into RNA, similar to remdesivir. But its mechanism of action is entirely different. So remdesivir is like a weak inhibitor that causes this barrier to replication, but molnupiravir is not causing a barrier at all. It is more causing replication errors so that the polymerase makes mistakes. And how does it do that? We found this happens mainly in 2 steps. In the first step, this active metabolite can be incorporated into the RNA, either instead of a C or instead of a U into the RNA. And then you have this compound in the RNA copy in the so-called minus strand. So the plus strand is the actual RNA genome of the coronavirus, and then this molnupiravir metabolite will be incorporated into the minus strand. But then there's a second step in the replication cycle where this minus strand is used as a template, and it will be copied again by the polymerase. And when this happens, then you generate a plus strand again, another copy, and that would be actually the copy of the RNA genome of the virus. But this molnupiravir moiety is actually causing errors because you can incorporate opposite of it, either G or A. So you will accumulate a lot of errors in the replicated genome so that the virus is actually not able to be functional and to leave the cell and to continue the infection. And we also made a molecular movie for how this works. This is now the second step of the cycle. You see the molnupiravir is already incorporated. It's already in the template strand, which is here in blue. And what we found is that it can either form a base pair with an A, then it forms 2 hydrogen bonds. But it can also form a base pair with a G base in the product strand. So this is possible because this component, the molnupiravir component, can actually adopt 2 tautomeric forms. And it can form 2 types of base pairs, either MA or MG. And that actually means that there's promiscuity and that there will be a lot of errors in the RNA product strand. Now, both of these antivirals have their issues. I talked about the issues with remdesivir, but also molnupiravir. It is effective to about 50% according to the latest phase 3 studies. Great Britain has just announced that they have approved for emergency use this compound, but it's still, we need still better antivirals, more specific ones, more efficient ones, and also, very safe ones with less side effects. And so what we are trying to do now as a next step, now that we understood how the available antivirals work and what their limitations are, we're trying to go for new classes of antivirals. And how do you do that? So, the antivirals I showed you are actually all belonging to this class of nucleoside analoga, and we would like to have totally different types of chemical compounds. And how do you find them? Well, you have to conduct a huge screen. You have to look through tens of thousands, hundreds of thousands of chemical compounds and find new inhibitors that are entirely distinct. And so how do we do that? We developed an assay based on fluorescence that allows us to detect the activity of the coronavirus polymerase here by a change in fluorescence. And this kind of assay can now be parallelized, so you can use a robot to screen tens or hundreds of thousands of compounds. And that is exactly what we're doing together with the lab of Herbert Waldmann at Dortmund. They have this fantastic automated procedure where they screen thousands of compounds with a robot. And actually we have, after a few months of work, managed to find a few first substances that we have now validated in the test tube. And these are new compounds that can block the coronavirus polymerase. And so in the future, we think we will be able to test those for their antiviral potential. First, of course, we would go to cell culture, infected cells, and then we would hopefully soon move to animals and see whether they can also have an effect on the infection process or on the spread of the virus. And it would be just wonderful if we could show that some of these new substances, which are entirely distinct from those that are now entering the market and that companies have developed, if they could be shown to have antiviral potential and then further developed into drugs. That is our hope. And as you all know, when you start from scratch with new compounds, this is a long-term project. But just as a reminder, there will be new coronaviruses also when we successfully have fought the current pandemic with the tools that we have available. So with this, I would like to thank the people who have been involved. When when the pandemic hit Europe and we all went to home office, we had a Zoom conference in the group and we were thinking, what could we do to help? We're obviously not vaccine developers. We, are polymerase experts. We actually work on RNA polymerase II, the enzyme that makes messenger RNA in cell. And we thought, well, we can probably use that knowledge to generate new knowledge on how the coronavirus replicates, and then we can use that knowledge to understand better how antivirals work, and then also to hopefully discover new substances with antiviral potential. And this is, what we are trying to do. So I thank all the people who have been involved. Here on top is the team that 1.5 years ago, within a record time of a couple of weeks, was solving the structure of the corona polymerase, actually in a race with 3 groups in China that had a jump start, as you can imagine. And all 4 groups in the end published around the same time in the same months in spring 2020. And then Jana and the 2 Florians joined now in the understanding of molnupiravir and also in our collaboration with Claudia Höbartner. That is extremely important. She's an RNA organic chemist, and she actually prepared some of those RNAs that we used for structural studies to lock the polymerase in defined states. And now we have this collaboration with Sonja Sievers and Herbert Waldmann at Dortmund to screen for new compounds. And hopefully I can come back one day to Berlin and present the antiviral potential of those, and that would be wonderful. Thank you very much for your attention.
Thank you very much, Patrick. And I think it's a perfect example of what we were talking about before, standing on the shoulders of giants. That's a quote I stole. It's not my quote. It's from Isaac Newton. So now Christiane will come up on stage and will tell us a bit more about the development of the technique, because obviously it was there when the pandemic started. It made the research of Patrick possible. So we hear a bit more about that, and then we can discuss the drugs and everything.
Yes, thank you very much for the invitation. And of course, there is a lot of techniques Patrick has to use for his fantastic research, also his decades, years of experience with RNA polymerases, but cryo-EM is just one of the aspects. And can I have? Okay, thank you. And in my talk, I just want to highlight the development of cryo-electron microscopy to become a powerful lead technique in structural biology to facilitate such structural work. And if we want to understand how a machine works, it's not enough to just know the components. We have to know how these components are assembled in 3 dimensions, and we require to understand the functional states of this machine. And the same holds for the macromolecular nanomachines of our body. And here in recent years, cryo-electron microscopy developed into a technique to really obtain such 3-dimensional images of macromolecular machines. To solve a structure with cryo-electron microscopy, we have to first isolate the complex. Then there is a vitrification step. The complexes are embedded in a thin layer of amorphous ice. There is cryo-electron data collection, and we have the raw images. It's called microscopy, but with the results from the microscope, you cannot do much because you have just noisy images. And then the real structures are coming from the 3D reconstruction, the single particle approach, where we need a lot of computational time then to calculate average 3-dimensional structures from the projection images from 10,000 to 100,000 of projection images. And then the fun part starts where we then do interpretation and model building. And cryo-EM became more and more powerful, and in 2017, Jacques Dubochet, Joachim Frank, and Richard Henderson received the Nobel Prize in Chemistry for their pioneering work and for inventing these techniques. And to show you the progression, I mean, sometimes it takes quite some while to make breakthroughs. I'm showing you here 4 structures of the eukaryotic human ribosome as an example to see how the capabilities of these techniques evolved. And around 2000, it was still state of the art to have resolution of 15 to 20 angstroms. And then with the advent of better microscopes, it was possible around 2004, 2006 to reach sub-nanometer resolution. And then with yet another hardware improvement, yet another new generation of microscopes, and importantly, the advent of direct electron detectors as camera systems, it was possible to break this important barrier of 4 angstroms, to have near atomic resolution. And this is so important because at this resolution, this facilitates de novo modeling of atomic structures based on the density alone. And this is not the end of the development. I mean, this was always called the resolution revolution, but it's more an evolution. And now we can, with ribosomes, reach about 2 angstroms resolution. And you see that there we have chemically very well defined the basis and the amino acid side chains. We can analyze chemical modifications. We can see small molecular bound ligands, and we can see magnesium ions and water molecules. And the hardware development was not the only reason for the progress. As you can see here, the fast computers which developed allowed us to use more and more images in the reconstruction process, larger data sets, and the pixel size became smaller. We could process larger files to see more details. And also the image processing algorithms became better and better. And very important was the development of multi-particle refinement techniques to sort the image data into more homogeneous substates. This is important for improving the resolution on one hand, but it also will provide you a lot of more information, because from one sample you can solve not one structure, but many structures. And here you see an example of the human polysomes actively translating ribosomes, where we were able to resolve 11 different states just from one sample, and we can cover nearly the whole elongation cycle with this. And currently we are using cryo-electron microscopy in a time-resolved manner to look at the in vitro assembly of the bacterial 50S subunit. And this is the result from the 3-minute time point, so just one sample from one time point. And you see how many structures we are getting here from a small nucleus where we see only one domain of the ribosomal RNA, and then it progresses to the nearly complete 50S subunit. And Patrick has shown you already that cryo-EM now, with these new capabilities, can be also used as a tool in drug discovery. And here is an example from our lab where we explore the assembly of the ribosomal 50S subunit as a target for new antibiotics in collaboration with Ralf Hoffmann from the University of Leipzig. And we are focusing on apidacines, proline-rich antimicrobial peptides derived originally from bee. And here we see this peptide sitting in the tunnel of the 50S subunit. And another example comes from the lab of my colleague Patrick Scherer from the Biophysics Institute. He is an expert in GPCRs, and he and his team, also with some collaboration from the Institute of Pediatric Endocrinology, they recently solved the structure of the melanocortin 4 receptor, MC4R, in complex with the G protein, and in the presence of the drug setmelanotide, which you can see here. And now with this work, they understand how this drug helps patients who have a mutation in the MC4R and suffer from obesity, how this drug then binds to the GPCR to activate it, to spur signaling, to do appetite regulation. So I want to thank the people involved in this work, Franziska Bichert, Justus Lörker, and Annette Unbehauen were involved in the 60S subunit at 2 angstrom resolution. Rainer Nicolai, Bo Kin, and Simon Lauer are involved in our ribosome assembly works. I already mentioned the work of Nikolaus Haider and Patrick Scherer in the Scherer group, and collaboration and funding. And I would like to end with a quote from Marcel Proust. The real voyage of discovery consists not in seeking new landscapes, but in having new eyes. And this is the perfect quote for electron microscopy. You just have to replace new eyes with new electron microscopes and faster computers.
Thank you, Christian. And Patrick, would you please come on stage so we can discuss this? And to say that, one of the mentioned researchers here is here in the room, Patrick Scherer. He will join us on the optogenetic panel later. So, would you like to start with asking questions?
Yes, I mean, Patrick, you are now using the high throughput approach to try to find new drugs. Have you also thought about going the classical way of rational drug design, using your beautiful structures to see whether something can be improved there?
Yeah, that's a great question. First of all, thanks for explaining so nicely how cryo-EM works. It really is the game changer also for us in this project. But the drug design, I think, is still a valid approach. So our original idea was, once we know how remdesivir exactly sits in the active site and how molnupiravir makes the base pairs and so forth, can we probably suggest small changes in the compound, like to add a few atoms here or there to make it better. And actually, Claudia Höbartner, our collaboration partner, is trying to do that, to, probably come up with an idea, how can you make a better remdesivir that blocks the polymerase more efficiently? I think it will not be easy in the end, because as you know, it's not just how the drug interferes with the target, it will have all kinds of consequences for metabolism, So it will be difficult. Once you start changing the compound, it will be difficult. But it's a second approach that we're trying, yeah.
I have a question in this direction.
Okay, would you please, Peter Hegemann?
I think the rationale about your approach is that if you use nucleotide isomers, you certainly inhibit the polymerase of the virus, but on the other hand, you also inhibit the polymerase of the human body. And the only way to go is to have specific drugs that inhibit the polymerase of the virus, but not the one of the human being. So in your screening assay, you should do a differential screening, right? So screen positively on your viral polymerase and negatively on the human. Is that done in this direction?
Yeah, that is fantastic. Thanks, Peter, for saying this. That is exactly what we're doing. So since we have all the cellular polymerases, I didn't have time to go into such detail, but since you mentioned it, we have the cellular polymerase, we have the human mitochondrial polymerase that actually often is causing the side effects, right? Because it will take those nucleoside analoga. And we have RNA polymerase 2, we can also make RNA polymerases 1 and 3. And we are testing them, are they inhibited by these compounds? And thank God, one should say, at least the nuclear polymerases, they have a much higher fidelity. They will not normally accept such, nucleoside analoga. But the mitochondrial one is quite vulnerable, and that is an issue. And with molnupiravir, actually, because it's so similar to the natural nucleotides, it will also be used even by nuclear enzymes, and that causes also some, concerns about if you would give that drug for longer, what could happen. So far, safety issues look good, but in cell culture, we know that molnupiravir also can be incorporated into DNA. There's a publication on that. So that's exactly going in this direction. We have to counter-select the substances that block also the cellular enzymes. And I think by going blind, and looking what's out there in chemical space, we may have a very good chance to find something orthogonal that would only block the viral enzyme. And that is our hope. And that should hopefully be much more selective and with less side effects than compounds that are currently used.
Okay, so I don't know who was first.
So, thanks for sharing the data. So along those lines, if you look at polymerases from other coronaviruses, I mean, we have 6 other coronaviruses, do you see also blockage of those polymerases? And wouldn't that be a possibility to test them even in patients? Because with the other coronaviruses, they are more harmless, and you could test your polymerases or your inhibitors whether they would block the coronaviruses, which are not so harmful.
Yeah, that's another great question. These polymerase inhibitors in general often inhibit even polymerases from different viruses, not just within the coronavirus family, but even different RNA viruses. And the reason is because they are substrate analoga. Once we have our new inhibitors, I think they will be much more selective because they will probably go, to pockets outside of the active site that are specific for certain viruses, viral polymerases, but even for subtypes. I think we have looked actually at the conservation of this enzyme between SARS-CoV-1 and SARS-CoV-2, and it's so highly conserved that even such pockets that are close to the active site but outside the active site, so allosteric pockets, they probably will be similar in other coronaviruses, and then what you suggest will maybe a possibility.
Maybe also good for MERS, right? MERS is very deadly, right? So that would be
So I think it was published,
Yeah, in principle, this is a general approach for this, right? Maybe you can also block other viruses, not only coronaviruses. Is it highly specific, remdesivir, or maybe you can use it also for totally other viruses?
Yeah, that
Ebola,
relates
for
to the
example.
question
Yes.
we are just discussing. The nucleoside analog are very broad. That's why, when the pandemic hit, the first thing companies did, if they had something developed for Ebola, which was remdesivir, they were trying it. If they had something for influenza, they were trying it, like it's the case for molnupiravir. But I think this will come to an end at some stage because of the massive side effects. And I think that's why, in the end, you need to have also specific inhibitors. But of course, it would be great if, it would also block other polymerases. That can be tested.
More question. So I saw you in the background.
Yeah, hello. Christian Hackenberger from the FMP in Berlin. I have a question maybe also related to the wonderful red square there. Which are the next walls to fall in terms of structural biology, especially when looking into different environments, like proteins in their, I don't know, native state, in cells, in lipid environments and whatever. What kind of methods and what kind of maybe also complementary approaches do you think are most important in the future?
I mean, this is a very easy question. The answer is cryo-electron microscopy, but not the single particle approach, but tomography, to spur in situ structural biology. And there are also really groundbreaking results with fit milling techniques developed in the Baumeister department in Martinsried. And Patrick just recently had a really breakthrough paper on methods development together with Julia Mahamid, where they observed the structure of a ribosome directly from the cell at 3.5 angstrom resolution with an antibiotic. And this is really fantastic. This is really a breakthrough, and this is a new milestone. And I think there is a future in integrative structural approaches and in situ structural work.
I see more questions coming, and I think we can spend a day with each of the breakthroughs. So I think we have to continue, unfortunately. I thank you, Patrick and Christiane, for doing this, being the brave first ones. And so in the background, there's Street and Mattes dealing with the microphones and with everything. So what do we do now?
Keep your microphones and just
So thanks for this and thanks for the two. I think we need a new slide. Okay, there it is. So, next topic. It's called Breaking the Wall to Information-Rich Diagnostics, and what is this about? So, it's about CRISPR-Cas and the gene scissors, and maybe you have met Emmanuel Charpentier this morning downstairs. And this is a story about a breakthrough where exactly that happened. Oh, I did not expect this, but it seems important. So what is it? So please welcome Cynthia Sharma. She's the winner of the breakthrough this year, and or one of the 10 winners. She's a full professor at the Institute of Molecular Infection Biology at Julius-Maximilians-Universität in Würzburg. She's a biologist and obtained her PhD in bacterial RNA biology here in Berlin. And her lab studies gene regulation and CRISPR-Cas systems in bacterial pathogens. And when they try to find out more about Campylobacter jejuni, a bacteria known for causing gut infections, they stumbled about something they didn't expect. This finding led to her breakthrough. She now will be talking about a technique which might make diagnostics simpler for us, called Lickert. And the team started off with COVID-19 as a proof of principle. Her matching partner, who the Charité invited, is Professor Dominic Neopak. He is assistant professor at the Department of Biology and Center for Synthetic Biology at the Technical University of Darmstadt. So in his previous work, he used optogenetic approaches to find out more about proteins. His lab still uses them as well as the gene scissor CRISPR-Cas. And Dominic will give us an oversight on the different methods and technologies based on the breakthrough of CRISPR-Cas. And before going to Darmstadt, that's the affiliation with Berlin and the Charité, he was based at the university in Heidelberg and working with Professor Roland Eils, here in the front, who is now the founding director of the Berlin Institute of Health Digital Health Center here at the Charité. So welcome everybody, and please, Cynthia, present your slides. The clicker is here. So, okay.
Should be okay, right? Can you hear me? Okay, so good morning. Thank you very much, Christiane, and also to the Charité Foundation for the invitation. So my name is Cynthia Sharma, and I'm a microbiologist working at the University of Würzburg. And it's really a great pleasure to be here, and it's really an amazing science summit. And I feel very honored to be selected as one of these winners in life science this year, together with my colleague Chase Basel from the Helmholtz Institute for RNA-based Infection Research in Würzburg. And in my talk, I would like to tell you about a new diagnostic method that we developed, which we think can break the wall to information-rich diagnostics. So diagnostics have really taken a front stage during the ongoing pandemic. So probably everybody of you has used at least one of these types of tests, maybe even this morning before you came to this event. And this tells you whether you have COVID or not. But often you're left with many additional questions like, do I have one of these variants? Do I have a completely different respiratory virus? Or do I have something completely different? So the challenge with these tests and standard diagnostics is that they typically give you one piece of information. But getting more information and being able to test for multiple biomarkers in parallel is crucial to make better personal decisions and also better decisions about healthcare. And as Christiane mentioned, we have developed a new diagnostic methods that we term Lickert, which is based on CRISPR, which can address these many questions and test for multiple biomarkers in parallel. And this method was developed in a very unexpected way based on a surprising discovery that we made while studying how CRISPR works in a bacterial pathogen. So CRISPR is now known widely as this powerful gene scissor, and Emmanuel Charpentier and Jennifer Doudna were awarded the Nobel Prize in Chemistry last year for the development of this revolutionary genome editing tool. And hereby, the Cas9 protein, which is a nuclease, uses RNA molecules as a guide to be directed to a matching DNA target, which is then cleaved. And as these RNA sequences can be modified, the whole system is mediating a programmable genome editing. And now there are many, many more applications of these different CRISPR systems, but I hope you can appreciate that originally CRISPR-Cas is an RNA-based immune system of bacteria and archaea and protects them against viruses. So also bacteria can be infected by viruses. And in this case, the CRISPR nucleases cut and inactivate invading nucleic acids from a virus, for example. So in these systems typically consist of multiple Cas proteins, like the Cas9 nuclease, which then bind to one of these CRISPR RNA guide molecules, which directs them to an invading nucleic acid from a virus, which is then inactivated. And typically, these RNA guides are encoded in a so-called repeat spacer array. So this is basically a library or a memory of previous viral infections. So once a bacterium is infected with the virus, a piece of the genomic DNA of the virus is incorporated into the system, so in the genome of the bacteria, and then in a subsequent infection can be used as a guide to direct the system to inactivate the virus. And it was for long assumed that these RNA guide molecules are always associated with the system. But when we studied the CRISPR system in the foodborne pathogen Campylobacter jejuni, we identified an unexpected new type of such RNA guide molecules, which are actually derived outside the system and have nothing to do with the immune function. And this is what we termed non-canonical CRISPR RNAs, which became the basis for this new diagnostic platform. So how did we find these RNAs? So Campylobacter is a human pathogen. It's the leading cause of bacterial foodborne gastroenteritis. And Campylobacter has such a native Cas9-based CRISPR-Cas immune system to protect itself against viruses. And the hallmark of these Cas9-based systems is actually another RNA molecule, which is called tracer RNA, which we have originally identified together with the lab of Emmanuel Charpentier while studying RNA biology in the in the human pathogen Streptococcus pyogenes. And this trans-encoded RNA, so tracer RNA, can bind to a precursor RNA, which is transcribed from this library of guide RNA molecules, and then together with the host factor RNase3 mediates processing and generation of the mature CRISPR RNA guides. So we are interested in how this system works in the bacteria. And especially we are interested also because there are emerging evidence that these systems have additional functions outside as an immunity system and maybe even impact on gene regulation. So what we did is we performed an experiment, so I forgot to mention, so for example in these genome applications, the CRISPR RNA as well as the tracer RNA are fused to a single RNA as a single guide RNA. So we are interested in how these systems work in the bacteria themselves. So we performed an experiment that we term RIP-seq to identify which RNA molecules are bound to the Cas9 nuclease in the bacteria itself. So for this, we added a tag to Cas9 in Campylobacter so that we can pull out the protein from the cell. And then using deep sequencing, we can analyze all the RNAs that are bound to Cas9 on a genome-wide manner. And this, as a positive control, revealed all the CRISPR RNAs and tracer RNAs of the system bound to Cas9. But then, surprisingly, we also saw that several other RNA other RNA molecules from the cell that were derived from messenger RNAs, which have nothing to do, in this case, with the immune system, were bound by Cas9. And actually, these fragments had a similar size to the CRISPR RNA guides and also showed a potential base pairing complementarity to the tracer RNA. So they somehow look like CRISPR RNAs. So we proposed that, similar to the canonical biogenesis of CRISPR RNAs based on binding to the tracer RNA has some kind of flexibility and can also base pair to other cellular RNAs, which then leads to generation of such RNA fragments that we term non-canonical CRISPR RNAs, which associate with Cas9. So these RNAs look like CRISPR RNAs, so the question was, do they also act as CRISPR RNAs? And based on a variety of experiments, we could show that indeed these RNA fragments can guide Cas9 to a matching DNA target and then mediate cleavage. So since we had observed that these non-canonical CRISPR RNAs can be generated based on binding of tracer RNA to cellular RNAs, we were wondering if we could maybe change the tracer RNAs in a way so that they do not only bind to CRISPR RNAs, to other RNAs from Campylobacter, but that they could hybridize to any RNA that is of interest to us and convert it into CRISPR RNA. And so we termed these changed tracer RNAs reprogrammed tracer RNAs, or short raptors. And indeed, this worked. So we did a lot of mutational analysis, and this showed that we can actually change the whole sequence of the tracer RNA part that normally hybridizes to CRISPR RNA repeats, and still we can generate functional CRISPR RNAs from any cellular RNA of interest, and then direct Cas9 to a matching DNA target. So previously, typically a guide RNA is provided to Cas9, which then is directed to a matching DNA target, which is then cleaved. But now with the addition of the reprogrammed tracer RNAs, we can convert any RNA of interest into an RNA guide. So basically, if an RNA is present in our sample, this leads to a sequence-specific cleavage by Cas9 of a provided DNA target. And the very interesting thing about Cas9 is that it can mediate sequence-specific cleavage. So based on this, we are able to detect multiple RNAs in parallel. And this actually became the basis of our diagnostic platform, Lickert, or short for Leveraging Engineered Tracer RNAs and On-Target RNAs for Parallel RNA Detection. So I think it's more complicated to say than cryo-EM. And so basically we start with a pool of RNA molecules from a patient sample and provide purified Cas9, as well as our designed raptors and a matching DNA target. And then by monitoring which of the DNA target is cleaved, we can trace back which RNA of interest was present in our original sample. So to first set up this Lickert assay, we use an in vitro assay where we can add commercially available Cas9 from Streptococcus, as well as synthetic raptor RNAs that we generate, and then use a set of different size DNA targets, which we label with the fluorophore at one end. And then we can monitor cleavage events on a gel electrophoresis readout and monitor either the uncleaved products or resolve the cleaved products upon cleavage. And we first applied or used these DNA targets for a set of different synthetic viral RNA fragments from different respiratory viruses. And here in this gel, you can see that the respective DNA target is only cleaved if the corresponding RNA of the virus is added, even if we have 3 or 5 different RNAs in the same reaction. So basically, with these reprogrammed tracer RNAs, we can detect multiple viral RNAs in one reaction. We have then also further expanded the Lickert approach by including a target-specific pre-amplification approach, and also changed the readout to a more practical readout using a bioanalyzer machine, and then applied it to patient samples that were previously tested positive or negative for SARS-CoV-2 using conventional PCR tests. And here you can see this first proof of principle experiment. So, of course, we also needed a control. So we designed a raptor against a human RNA-SP control RNA. And all of these 9 patient samples can be tested positive based on DNA target cleavage efficiency. Then as a negative control, we targeted an RNA from Campylobacter. And also these patient samples were tested negative for influenza virus. But then, most importantly, we designed a raptor against SARS-CoV-2 and then provided 2 different DNA targets, which are different by just a single nucleotide, which actually corresponds to one of the virus variants that is connected to higher infectivity. And all of these previously positively tested patient samples were tested positive for this D614G variant of the spike protein, indicating that this was the variant that was presumably spreading during that time when these patient samples were taken in Würzburg. But so these proof of principle experiments showed that Lickert can detect and even distinguish different viral variants at single nucleotide resolution. So to summarize, I've shown you that we really started from a basic fundamental discovery in microbiology while we studied the CRISPR-Cas system in a foodborne pathogen, Campylobacter, which led to the discovery of these non-canonical CRISPR RNAs. We then applied CRISPR engineering and developed these reprogrammed tracer RNAs, or raptors, which we then translated into a new diagnostic platform that can link the presence of an RNA of interest to specific cleavage by Cas9. So Lickert can really change the way of diagnostics, going from a few biomarkers to detecting multiple biomarkers in parallel. And while this detection of the respiratory virus was just a proof of principle, we see many more applications, such as detection of different pathogens or also SNPs, but also including cancer diagnosis or testing, for example, which antibiotics should be subscribed. So while we are still while we still have several steps to go to have a commercially available test, we really hope that Lickert can change the way of diagnostics and provide valuable information to doctors for making life-saving decisions. With this, I would like to thank everybody involved. So this was really a very fun collaboration with the lab of Chase Basil and his student Chanlei Jiao. So they were really the driving forces of the CRISPR engineering and developing this test. Then the non-canonical CRISPR RNAs were originally identified by Gaurav Duga, a former PhD student in my lab, and then further studied by Cynthia Sharma, a postdoc in the lab. And then to apply to set up the patient testing, we teamed up with our colleagues in Würzburg from the medical microbiology and diagnostics, Christoph Schön and Oliver Kurzei. And with this, I thank you for your attention, and I hand over to Dominik.
Yes, thank you very much, Cynthia. I'm really excited to be here, I have to say, and to give the matching talk for such a fantastic breakthrough. So let me see, these are not my slides. In this direction here. Are my slides coming actually when I click next, or aren't they? Should I click just through once? Yeah? Let me see. Okay. So can enjoy the slides once more. Wrong direction? Okay, let me see. But the red says, this was right? Okay. Well, I'll try. I think this is the correct direction. Excellent start. Sorry? Yeah, maybe it duplicated, yeah. Okay, let me see. Really? Oh, that's bad. Well, sorry? Ja, jetzt nicht auf dem Stick habe ich jetzt nicht dabei. Ich habe sie als PDF und PPT geschickt. Ansonsten mache ich das spontan ohne Slides. Okay, so I will try without the slides. That's fine. So I wanted to talk about CRISPR as a treasure trove for molecular biology. And I think it's really a treasure trove if you think about it in detail. It was already presented that CRISPR is an adaptive immune system, and this originates from a rapid evolution between phages and microbes. And this rapid evolution has a number of interesting consequences, because evolution is not very smart, but it has a lot of time, and therefore can be very innovative. So if you look at the different steps of the CRISPR-Cas immune system, you rapidly realize that the system has a number of traits that we can explore as technologies. One, it can adapt sequence information from the phage world and store this sequence information in form of a database. All right, so I'm already a few slides ahead. It can take this sequence information from a phage world and store it in a database. The CRISPR loci that were already explained. So it's a good system to adapt information and to store information. Second, it's a very potent sensor, which was also just explained. It can discriminate nucleic acid sequences at a single base resolution. And third, it's a very potent actuator, so it can cleave nucleic acid sequences, which we can use to perform genome editing. But CRISPR even has more traits that we can explore that results from this rapid coevolution between phages and bacteria. One is that nature had to come up with a sophisticated regulatory system in order to keep the CRISPR-Cas immunity in check, because otherwise phages would probably not exist anymore. And second, CRISPR also determines how information is propagated throughout time and throughout populations. And we can actually explore all of these different traits, and presumably there are many more, as technologies, and actually develop them into technologies. And the first is very obvious. I don't need to explain it at all, that CRISPR is of course a gene scissor which we can use to edit genomes, and this revolutionized biology, biotechnology, and is about to revolutionize medicine. But also simply using CRISPR as a detection system, as was explained, has very huge potential. And I just want to point out that this field of CRISPR-based diagnostics is still pretty young. It's just about 5 years old. It started with a number of papers published in 2016, 2017, and 2018, most prominently two systems called Detector and Sherlock. So these systems are based on CRISPR-Cas12 and CRISPR-Cas13, two other enzymes from the CRISPR family. And they have a very interesting property, which is when they bind to a specific target, which is complementary to the CRISPR RNA, their nuclease function will carry out collateral cleavage. And collateral cleavage is nothing but a nice scientific term for saying that the nuclease function goes crazy. So these enzymes will basically start to look around and just chop up every single-stranded DNA or RNA that they can find in a sample. And now one can use this property of collateral cleavage to build also CRISPR-based detection systems by basically designing probes comprising of a single-stranded DNA or RNA that are fused to a quenched fluorophore, which will be released upon cleavage of these single-stranded sequences. And these systems are highly sensitive. They're relatively rapid to set up, but the downside of these systems, their major limitations, was that they were difficult to multiplex. And this is now overcome by the breakthrough that Cynthia presented with this Leopard system, which uses engineered tracer RNAs as a kind of fishing rod, actually, to fish specific RNAs from a sample, and then to build an active CRISPR-Cas complex that targets a specific reporter, which is specific for every RNA sequence. And this is not only sensitive, but also easy to multiplex. But I want to zoom out even further. So we can also use the CRISPR-based adaptation, so the ability of CRISPR to store information in the genome of microbes in order to engineer tools that we can use. For instance, CRISPR-based recording devices, which you can use to store transcriptomic information in the genome over time. So you have basically a transcriptional record in the bacterial genome that you can then read out by DNA sequencing to identify how a cell behaved at a certain point in time, whether it encountered certain toxins, what kind of environment is encountered. And this is also interesting, again, for instance, to build diagnostics that we can swallow and that will travel through our guts, and at the end we can read them out to see whether in certain parts of the gut we have infections. My own lab is working on these CRISPR regulatory layers called anti-CRISPR proteins, where we apply protein engineering as well as genetic engineering strategies in order to turn these regulators into something which we can use to control CRISPR-Cas enzymes in human cells. So we can combine this regulatory layer with optogenetics in order to be able to activate CRISPR-Cas activity in selected cells at selected time points. But we can also combine these regulatory layers with endogenous cellular signals to discriminate different cell types and making sure that CRISPR is only active in selected cell types, maybe in the future in human patients. And finally, this was also already mentioned in the panel this morning, the ability of CRISPR to determine how information is propagated through populations throughout time can be harnessed in form of CRISPR gene drives that actually make sure that when you introduce a mutation in one allele of a diploid organism, this is copy-pasted also to the second allele. And thereby you can basically circumvent the classical rules of Mendelian inheritance that we all learned in school and make sure that a mutation that we introduce is actually propagated rapidly into all the progeny of a population. And this you can use to actually engineer populations. Now, I would like to leave you with these images. CRISPR is a multifold of different technologies, all of which we can harness for different purposes. It's of course on us as scientists, as society, to make sure that these are developed in a responsible manner, and I think we can. And the example that Cynthia showed really excites me a lot about the future of this field, and I think it shows the huge potential CRISPR-Cas has in many different application scenarios. Thank you very much.
So Dominic, I'm sure you have questions you want to ask Cynthia. Would you like to start?
Yes, so absolutely. I'm actually pretty excited about these non-canonical CRISPR RNAs. Do you already have ideas, hypotheses of what these non-canonical CRISPR RNAs do in nature?
Yeah, so this is a very good question. That's actually how we initially started, right? Because we wanted to find out what the system is doing. But even for Campbellmarkt itself, this was completely unexpected. So even for CRISPR biology, these non-canonical CRISPR RNAs are a new concept. But we still do not know if they, for example, have a function in gene regulation, or maybe they could limit horizontal gene transfer. But it's also possible that this is maybe just a side effect, an off-target effect of tracer RNA during this immunity system function. So this is something that we are still further exploring.
All right, and unless there are already questions from the audience, maybe a second one. How are you planning to develop this technology further? Are you planning to commercialize it? Are you already in the process of thinking about clinical approval at some point? So can you tell us a bit about what are the next steps that you are now heading for?
Yeah, so I mean, at the moment we are still at the very beginning, right? I mean, I just showed you these proof of principle tests, and we still have this quite a bit complicated readout with DNA cleavage and bioanalyzer or gel electrophoresis. But definitely we are further pursuing this process of developing a commercially available test. And so we are doing different optimizations of the method now, so including also methods to improve the sensitivity, for example, also by trying to use different nucleases in this Lickert setup. And also different readouts. For example, instead of a gel-based readout, we are trying to set up, for example, a microarray-based readout, which would really facilitate this multiplex aspect of Lickert. Or many of these CRISPR-based diagnostics are based on lateral flow assays. So this is also one thing that we are now pursuing to further develop the method.
Exciting. Maybe, there's
one question, Shamreen, to please. Thank you.
Can you explain it a bit how it would work when you look for SNPs, for example? Because I think this is also a very exciting potential to look to SNPs.
Mm-hmm. Yeah, so if the SNP would be also transcribed into an mRNA, so the detection of a point mutation is based on the DNA target. So I only briefly mentioned this in the end for this coronavirus variant, but in principle you can develop or design a raptor that recognizes a specific transcript, and then you provide two different DNA targets and put one point mutation in the seed region of the CRISPR RNA. And the system is so sensitive that it would only bind if the sequences at the seed has a full complementarity match. So it would only cleave the respective DNA which has the corresponding point mutation.
And this is also possible without blood, right? in principle?
Yeah, I mean, this is another thing that we still have to further optimize. So I didn't mention it, but for example, for these patient samples, we still had to do an RNA extraction and then apply the Lickert. But this is also one thing that we further optimize the sample preparation. Like, I mean, ideally, later on, we could take a blood sample and then do detection.
I think Peter has a question.
I remember an assay developed by Feng Zhang by using a similar approach by using this Cas12, and he has a multicolor fluorescent assay that he can directly detect.
Yeah,
Very
so
similar
this is one of these Cas12 or Cas13 based diagnostics, and this is typically based on this lateral flow assays at the moment, so that you can also provide a point of care diagnostics. And they also did some multiplexing, but this is based on using different Cas12 or Cas13 variants from different species. So I think they have up to 4, if I remember correctly, but they cannot go to many more samples in parallel.
Okay. But he's also using you say your Cas is activated by a specific RNA, and then it's continuing off target.
Yeah, there's. Yeah, so there's a little bit different. So there, the nuclease is guided to a specific target DNA or RNA that is present, and this then activates a collateral RNase or DNase activity, which can be used then, for example, to cleave a reporter, like where you have fluorescence molecules that are quenched. So based on this, you can only have one piece of information. Yeah, if you can use, for example, up to 4, but you could not go to hundreds or thousands, right? Yeah.
I can't see your first name, so it's Mr. Besser.
Besser, yes. Thank you very much. What I missed, maybe I didn't understand it correctly, but I saw that in the original experiments you showed us that there are a lot of off-target effects and a lot of not correct bases were detected by the molecule. Now later on, when you looked at the SARS, you showed us that you can be precisely looking for one variant and not see the other one. So how can you make it so precise then later on, where you have a very unprecise system with off-target effects in the beginning?
Yeah.
Maybe I missed that.
Yeah, sorry, maybe this was confusing. So these are 2 different types of base pairing interactions. So the one where we see flexibility and this imperfect complementarity is at the level of tracer RNA binding to an mRNA to generate a CRISPR RNA. But then this CRISPR RNA has a specific guide sequence, and there you have a 100% match, and you also need a PAM sequence. I mean, the ones who like you need also for genome editing. So there you have then really high specificity of targeting to this provided DNA target.
One last question.
Yeah, thank you so much. Julius Uppmeyer, I'm with the Digital Health Center here. I was wondering if you could dive a bit more into the limits of the multiplexing, because you've just teased that you can sort of go beyond the 4, but sort of where does it stop, and what are the limiting factors for that?
Yeah, I mean, this is a good question. So, I mean, we are still in the process of further developing this method. But in principle, our vision is that we could really, if we use microarrays, for example, as a readout, we can test hundreds or thousands of RNAs in parallel. So for each RNA of interest, we could design a raptor and then have specific targets on the microarray and then use this as a readout. I mean, microarrays are not used anymore for sequencing so often, but I think a lot of diagnostics labs still have microarray readers, so this could be a practical readout for this. But yeah, and I think, I mean, for example, one thing that we are also looking into now is, for example, diagnosis of sepsis, which is really a problem in the clinics, because very often then people come with sepsis and it's not known what is the pathogen that causes the disease, and their time is really crucial. So if we can develop a test, ideally on site, that could immediately test what kind of pathogen and also which kind of resistance marker is present, we could maybe really develop, hopefully, a life-saving diagnostics. But we are still far away from going to this.
Thanks. I think it's a beautiful piece of translation we would have then if we had a test for sepsis. So now it's a networking coffee break. We meet again at, I guess it's 12, to proceed with 1 hour with José-Alain Sahel, Peter Hegemann, Oliver Zeitz, and Patrick Scherer. And let's see how we go with that. So enjoy your coffee. Thanks for having us. And see you later. Okay, what we do now is we are breaking the wall to restoring vision for retinal degeneration. So we're using optogenetics, and we talked about it this morning already for those who have not been here. I think there's a lot of beauty in this finding because it's really about translation, basic science into medicine, and finally helping blind patients. It's an interdisciplinary approach between algae protein research and neuroscience and ophthalmology, and about a finding in 1984 where somebody saw algae can see. So what do we do about it? And now we know what we can do about it, that algae actually can see. So welcome again, our breakthrough winner 2021 in life sciences, Professor José-Alain Sahel. So he's an ophthalmologist, a clinician scientist who developed a groundbreaking neuroprotective strategy to rescue central vision and first-in-man studies with electronic retinal implants, gene and cell therapies to restore vision in patients with blinding retinal dystrophies. He led the clinical trial documenting for the first time partial visual recovery after optogenetic therapy in a blind patient, and therefore is awarded the Breakthrough of the Year. He divides his time between the University of Pittsburgh, USA, and the Sorbonne in Paris. He is a founder of various biotech startups and has won many awards. And he is an officer of the Legion of Honor in France, which I think Napoleon Bonaparte actually founded about 200 years ago. So I think that's something to mention as well. It's spectacular. So we have an hour. We have 4 panelists this time. So we do it a bit otherwise than the other panels. We do have Professor Peter Hegemann. We already celebrated his Lasker Awards this morning. So he's a biochemist who has spent many years in research on proteins. And in Syracuse, New York, he worked with the physicist Ken Forster, who postulated in 1984 for the first time that algae have rhodopsins as humans and animals do in the retina and therefore can see. Peter came back to Germany, and in 2001, he identified the channel rhodopsin as an ion channel protein, which can be activated by light. So he laid important ground for optogenetics and life sciences. And first of all, Karl Geiserow and Boyden in Stanford used the channel rhodopsin to make a mouse run, a famous experiment you probably all are aware of. So since 2005, Peter is a professor for biophysics, and since 2018, a Hertie Professor of Neurosciences at the Humboldt University here in Berlin. He has won numerous prestigious awards, and he's still closely working with Karl Geiserow in Stanford. And through NeuroCure, a cluster of excellence, he is connected to the Charité. So welcome, Peter. Then we do have Patrick Scherer. He is a research group leader at the Institute of Medical Physics and Biophysics here at the Charité. His group works on protein X-ray crystallography and signal transduction. And in 2009, with Klaus-Peter Hoffmann, he described the signal cascade of how rhodopsin transmit information to the brain. So he's not focused on rhodopsin, but proteins of all sorts. Recently, he worked together with Peter Hegemann to find out more about the structure of a rhodopsin of another algae called Cocomyxa. I hope that's right. Okay. All these algae. So, and then we do have Professor Oliver Zeitz. He is an ophthalmologist, ophthalmic surgeon, and retina specialist based in Berlin. And Oliver's main scientific interest is translational medicine to overcome sight-threatening eye diseases. He joined the Charité in 2017, where he is leading the eye clinic at the campus Benjamin Franklin. And previously, he was the head of global clinical development ophthalmology at Bayer. And in that capacity, he oversaw a development portfolio comprising multiple late and early stage clinical drug candidates in ophthalmology. And the most visible program was aflibercept for intravitreal injection, which was approved globally under the brand name Eylea and 5 indications and established itself rapidly as a blockbuster drug. So Oliver continues to be committed to contribute to the development of new treatment options to prevent blindness, and he does that at the Charité now. So thanks for all of you coming. We do it a bit differently because we're 4. So I welcome José on stage for his presentation. And after that, Peter Hegemann and Patrick Scherer just take over, coming on stage. You stay here, and they do something here with a flip chart, actually, and have questions. So really low-key techniques. I think that's great. After that, the 3 of them will withdraw after, I think, half an hour. And then Oliver Zeitz will present some slides. And we open we have another vision. We look from a different angle, from the eyes of an eye doctor, actually. So welcome, everybody, and have fun.
So thank you very much, Christiane, and thank you to the committee that decided to include me into this program and to award the breakthrough. Having heard the first talks today, it's very humbling, so I'll try to be not too disappointing and try to talk about the work we did over the past many years, trying to restore some vision into blind patients. So if I can, I guess this is what I have to do, yeah. So first of all, I have to disclose that I'm founded several companies, and GenSight especially is developing this therapy. I also would like first to acknowledge that this work is actually the work of many people. I put a few pictures, especially both on Rosca. We have been working together for the past 15 years on many aspects of vision, and especially optogenetics. And Denise Alcara and Serge Picot in Paris. But also many people, as you will see throughout the talk, have been involved in many parts of this progress that we made, although we are still at early stages. So as a reminder, in retinitis pigmentosa, which is the most frequent genetic disease of the eye and the more complex genetically inherited disease, we are talking about 70 different types of causative genes. And for many of them, we talk about tens or sometimes hundreds of mutations that are causing the disease. The pattern is that this is leading to the loss of photoreceptors. Photoreceptors are located in the depth of the retina. The light is coming through the pupil, reaching the retina from this side and getting through the retina to reach photoreceptors. It's being absorbed at the level of photoreceptors outer segment, and we'll hear about seminal work on rhodopsin about that. And this is triggering electrical responses that are being processed, especially in the outer retina, temporarily into the inner retina, and the signal is then conveyed through the ganglion cells and the optic nerve to the brain. The real place where we see is the brain. Most of the mutations involved in retinitis pigmentosa lead to the loss of rod photoreceptors, the cells that enable us to see at night, and patients are first experiencing a loss of dark adapted vision. Then, over time, the cones, that are a minority of the cells, but the most important cells for central vision and light-adapted vision, start to degenerate, and patients experience what is called tunnel vision, where there is a narrowing of the visual field over time. They start with 40 degrees, they don't notice it too much, and then it becomes 20, 30, 20, and then 10, and at some point, because the eye movements are no longer able to compensate for that, they are experiencing a lot of disabilities. This tunnel becomes narrower and narrower over the years, and over the course of 15 years, sometimes less, sometimes more, they become fully blind. So this disease has been the target of many approaches. Maybe we'll discuss that later, especially gene therapies to correct gene defects, and this does include currently CRISPR-Cas approaches. But clearly, this is only possible when you are able to target the disease at the early stages, when the photoreceptors are still present, connected, and viable. If the photoreceptors are already quite advanced in degeneration, not even to speak about the stage where they are dead, there is no possibility to correct the gene defect. And this is where you need to define approaches that could be gene-independent, targeting the remaining cells in the retina to protect the remaining vision, or even better, restore the lost vision. So this is what the disease is looking like. Over the course of time, rods are degenerating, and patients are losing the rods and the dark adapted vision. Then the cones are degenerating. Then they lose progressively central vision, which is also color vision, and eventually there is nothing left. Optogenetics can be positioned at this stage of the disease, certainly not in the early stages, although there are approaches that we are working on that could be earlier, but still very early stage. So optogenetics was born many years ago, and we have the key inventor in the room. And really discoveries that were done in algae. I think this is one of these wonderful examples where we can tell people basic science is so important because nobody could fathom that this could eventually help anyone. And even the discoverers probably didn't think about that at that time, although I know that they thought about it. But the goal is to try to replace the very complex machinery we have in mammalian cells, where all these G protein coupled cascade with many proteins involved, the first of them being rhodopsin, and we'll hear from that from Patrick Scherer, is targeting an initial event through the absorption of one photon by the vitamin A derivative, and eventually over the course of a very fast process, there is amplification of a signal at the level of 1 million amplification, and this is triggering opening of a membrane, and this is triggering vision. Because many of these genes are involved in retinitis pigmentosa and other conditions in the eye, it's easy to imagine that if we could bypass that or replace that, this would be wonderful. Because these opsins were discovered and they comprise pumps or channels that can open the membrane or open the pump as a response to light, the idea came to maybe use that and to transfect the membrane in the remaining cells in the retina. And as you know, this has been used in many other parts of the central nervous system and beyond, to bypass, to really restore this lost cascade with a very rudimentary and simple mechanism. Clearly, we'll talk about that. You cannot replace all of that with just one protein. There is a reason why nature has developed all of that. It's really because this is such a sensitive system that can adapt at many levels of light, but at least it can restore some opening of membrane, some channels, and so, or some pumps. So the idea was to use this, could be the pumps like heterodopsin, and this is the work we started with Boton Roska many years ago, published in 2010, I think, trying to reactivate dormant cones in the retina, the cells at the stage where the photoreceptors have lost the outer segment but are still viable and are still connected to the innermost cells in the retina. The beauty of that is that these cones are connected to both the on and the off pathways and the many pathways of vision we have in the retina that are processing vision. But we found out that this could apply only to a minority of patients, and there were some complexities in targeting. It worked beautifully in the mouse, but once we tried to translate that to human retinas and to primate retinas, we found there were a few issues, but we are working on that. The other approach is to target the innermost cells in the retina using channel rhodopsin, the initial channel rhodopsin that was discovered by Peter many years ago, or optimized channel rhodopsin, and you try many of them. The cell type that could be targeted could be the bipolar cells. And again, in the bipolar cells, you could target the on or the off pathway, like the light or dark pathway, to be extremely oversimplistic. Or you could target even the amacrine cells, the cells that are targeting the spatial resolution. Or you could target the ganglion cells that are the innermost cells in the retina and are really forming the optic nerve. So this is the approach I'm going to describe today, although the other approaches are very valid, and we worked on many of these. So these are the many approaches that could be tried, and we worked on that, and on that, and this is the one that we were able to move into the clinic recently. When you look at patients' images of the retina, and there is a technology called optical coherence tomography, the ganglion cells are viable even at very advanced stages of the disease. So the idea was to transfect the ganglion cells because they are still present in most of the patients. This was tried by Pan many years ago in Detroit using the initial channel rhodopsin, but the amount of light you need to target it, to activate that is massive. So, and this is the work that Pan published many years ago, and I will skip on that just for the sake of time, not because it's not an important work. So we tried to optimize the technology, and first was the vector. the second the promoter, the third one was really the protein itself, and then the system to activate it. So first we had to find a way to move from the classical channel rhodopsin in the light which is very bright and the efficiency of light is extremely high in this wavelength, to something which would be shifted into the red, which would be less energetic and probably safer. So we tried that, and eventually we tried REACT, which was developed by Roger Chen, and we tried the Crimson-R that was developed by Ed Bolden at MIT. And we found out that Crimson-R was very efficient, although, as Peter may say, there are some flaws with it. We also tried to develop many types of vectors, and I will be quick on that. We tried different variants of AAV2 and trying to find how we could get the best transfection. Many technologies to do that, quantifying the expression using using GFP or TD tomato, also recording of the electrical responses, because the most important is there a response in these cells? And eventually we were able to optimize and to find the construct with AAV2, 7M8, channel rhodopsin, TD tomato, Crimson-R was the most efficient, as you can see on these graphs. So this is the one we moved into the clinic, but before we're doing that, we try to determine the response of a cell. So using multi-electrode arrays and also biphoton microscopy, we are able to record at the level of ganglion cells and to try to simulate the level of a signal. If you take different shapes that are projected onto the retina and you are trying to record the activities, you find patterns of activities. And this tells us about the coding of the information we need to provide to the cells to really get to something which would be as physiological as possible, not even to speak about talking the language of the retina. So we did that, and the combination therapy, after many studies in primates, was a combination of a vector with AAV2-7M8, this Crimson-R protein tagged to TD tomato to get to the membrane, coupled with a medical device with our goggles, because as I started to allude to that, we need a large amount of light to activate the retina, and we need also to adapt to many levels of light. In natural life, we are adapting to 11 log units of light, and these proteins, maybe I'll be corrected on that, are adapting to 2 log units. So there is something that we need to compensate. So these goggles have a camera in front of them, and this is a very specific type of camera, event-based, and there is a projector at the back, like what we have on the screen, sending the light onto the retina. The trial was a dose escalation study that we started in Paris, London, and Pittsburgh, starting with a low dose, then moving into a second dose, then third dose, and now we got approval to move into the highest dose because of the safety that we observed in this initial study. Because of COVID, the patient who needed to come back for training, only a few of them, actually one of them only could come back because there was a ban on clinical trials for anything which was not vital. So only one patient was able to do all the training. And now we have more patients coming back that I will report at the end of the week in the US. And but this patient, which is the one I was asked to focus on, was the patient that went through all the training and all the adaptation of the goggles in this trial.
On est en train d'observer dans l'écran, c'est des petits points qui correspondent aux événements, donc aux variations de contraste, qui correspondent typiquement à un changement, à une dynamique, donc les mouvements de ma tête, mes lèvres,
so they are not classical cameras. And as you will see, this is the CEO of one of the companies we formed developing these goggles, these cameras. When he stops moving, there is no image. So normally I'm supposed to do that on stage tomorrow, but I'm very scared because there is some flickering of light and it doesn't really work. But anyway, what it means is that these cameras are only functioning real time at any level of light, anytime there is a change in the signal. If there is no change, there is no signal. So the trial was a combination of a gene therapy and the goggle and the rehabilitation. So the patient was able, and I hope the movie will be working, to detect objects on the table, to quantify them. And we also performed in parallel multi-electrode recording at the cortical level to check that there was a signal corresponding to what the patient was seeing. So this was the first patient reporting any benefit of optogenetics. And so this is without the goggle, after the therapy. So the patient is scanning to try to find the object on the table. And he has some bear-like perception. So he could detect the edge of the table, but he couldn't see any object on the table. And so this is with the goggles, stimulating goggles. And so he has to adjust the goggle. If you see the screen, this is what the patient is seeing. The camera is seeing, not the patient, but the camera is filming. And so he's adjusting to a line. And at some point, he will detect the object. As you can see, it's not fast. And it's a real-time recording. and he gets something, and so he did that with different objects. We have different size of object, different contrast also, but all of that is in the paper. And eventually he sees some vibration corresponding to the camera, and he will align and then grab the object. Yeah. Okay. So this is a first report, and we also did the recording at the level of the cortex, and what we found is that in correspondence to this activation, there was a desynchronization of occipital oscillation corresponding to the classical pattern of vision, and this was absolutely above any chance level. So this is actually the quantification. Only the cortical area is activated, and this is far above chance level. So now we are getting more patients coming back with more training, and even this patient is using that at home now for daily activities, for example, to detect crosswalks, which was the first thing he reported. And the study is now moving into a last cohort with a higher dose, because this was the lowest dose that was tested. So it's part of a spectrum of therapies that can be adapted for this patient, some of them being gene therapy, some of them being neuroprotection, some of them being neuroprosthetics. But this first proof of concept is enabling us now to listening to the patient, develop better ways to stimulate the retina, and optimize the therapy. But I think I'm just reaching the end of my time, so I will thank everyone for the attention and go on to the next. Thank you.
José, come to us.
I can stay here. Yeah.
Yeah,
I think you must stay at the red.
Okay, on the red.
Yeah.
Thank you very much for the presentation, and I'd like to remind the audience where the channel rhodopsin comes from, and it is a very small alga which has two flagella, which are controlled, and here I should change the color. And here is a little iceberg, which was not so easily visible here. And this is a location where the channel rhodopsin is located. Yeah. This is where the protein is. And interestingly, what happens if you stimulate these cells, you get a first photoreceptor count and then a flagella count, so which means first ion influx here and then an action potential, which is this one here, in the flagella. So the principle is very old, about 3 billion years old, and certainly it took almost 3 billion years to discover this. And then we began to work with this in about 85, but it took us 15 years to identify the photoreceptors, and that was in 2001. And the first step we did to get attention from the neuroscience is we expressed it in human embryo kidney cells, and were able to activate the kidney cells and depolarize it. And then Tuopan did the first experiment in mouse that was mentioned by José. And then after these initial trials, we decided to organize a Dahlem conference here in Berlin in 2012. And one of our special guests was José, and he explained us the preliminary experiments he did with Boton Rosca on these issues, and everyone was very skeptical. So this is normal in science. If you go into a new direction, first they don't believe you, and when it works at the end, then everyone is happy and says this is not so surprising, because if it works in algae, why shouldn't it work in human retina? So it is very interesting, and I'm very pleased to see that the first patient has been treated, and my congratulations to José about this success today. And if you remember that many different labs in the world, at least 4 in the US and 1 in Japan, were trying to get this to work, it is certainly a very special moment for me and all of us in the optogenetic field to see that it worked finally in Paris. So now comes the next question. So if you do something in science, then the demand increases certainly, and everyone wants to increase vision. And my first question to José is, what is the next step to improve light sensitivity and spectral resolution? Because what we expected is, and we had a discussion about this in 2012, with Ebrard Cramer, who presented his work with the chip technology. It's an electronic chip which is implemented behind the retina. And the difficulty with that is the connection between the electronics in the cells and what we expected at that time already, that optogenetic applications might have the advantage to improve spatial resolution. Jose, what is your opinion about it?
Well, thank you. So I think there are different ways to work on that. The easy one, the low-hanging fruit, is that now the patient have the proteins expressed in the retina, and the goggles and the stimulation device is outside the eye. So we can modify anything, other parameters, and from the physiology of the visual system, and also from the feedback the patient is giving us. And I showed you, we have a couple of papers on computational neuroscience mimicking the signal, trying to calculate actually what type of resolution you could achieve. There is probably a way to optimize the existing system with just the stimulation system, and this doesn't require any injection, anything to do to the patient. But the next step is certainly maybe having a protein that has an even better kinetics, although this one is pretty good, but I think there's still the more we'll get into the red and even close to the infrared, we might be able to combine that with remaining vision. Currently, we are treating blind patients. One of the patients I'm going to report at the end of the week was totally blind, and now she's able to grab objects and everything. So, but this patient, certainly there is nothing that could interfere with her natural vision. But other patients still have some remaining vision, and you don't want to compromise any of that. So the more you would get into the infrared, the better it would be to be able to combine the enhanced vision and the vision that is still remaining in this patient. And we have another approach to combine that with neuroprotection to keep alive the neurons that are still functional. But the main thing is really to be cell-selective, cell-specific. So this is why we spend so much time on the vectors and now on the promoters. Last week, we just published a big paper on a very systematic development of vectors that are cell-specific in the brain and in the retina, because if we can be cell-specific for ganglion cells, instead of activating all of them at the same time, which certainly is not the natural way we see, but targeting specifically one type of on cells or off cells, especially the central ones, then the resolution should be improved very significantly. And this is why the bipolar cells or the cone cells was initially very appealing, but not very easy to translate into humans. This one was easier to translate, doesn't mean it's the best. And the other thing is really to work on the threshold of stimulation that we're going to use in these patients. Currently, we believe that we are probably above the threshold we need, so just to adjust that to something which would be more physiological. But I'll stop that. The stimulation system is also a way to optimize it.
To go on this side, maybe the threshold for cones is very high and your system low. Why is this the case? Why do you need not so high threshold for your system? So the level of light we need in these retinas is very high. It's very safe. We checked that it's far below the threshold for safety, which was a big issue with the initial channel rhodopsin. We are far below, but still we need to amplify the level of light several log units to get into something which would be a stimulation. So we are far above the classical cone vision. As you know, several groups are trying to develop channel rhodopsin that would be working in natural levels of light. Probably a good idea. I still think that the adaptation to the changes of the level of light are going to be an issue. What we were very concerned in this patient would be photophobia, and they are not photophobic. And if you have something which is responding to natural light, maybe the patient may not be so happy with the just being blurred with artificial vision. Do you think it is possible to also to put inside anion and cation channels in parallel, maybe also for on and off targeting?
Yeah, I think it's a great idea, but in the retina, the on and the off pathways are relying on different cells. I was very intrigued by the introduction we got from the Charité dean about cells, and the retina is really specializing the signal between the on and the off and the on-off. So having that in the same cells may not be the best, but if we have cells-specific promoters or vectors, which we are starting to have. I don't have time today to speak about that. Then we could do that. It could be on one and off on the other one, and we could enhance the receptive fields in the retina using this approach. But it's probably the next generation, not to talk about color vision.
Yeah, but I think at the moment we are in a better situation than a couple of years ago because we have anion selective channel rhodopsin and and potassium selective channel endorbsin. So if you express these in off bipolar cells and the sodium sensitive and on bipolar cells,
exactly.
you can probably create a better sensitivity and a better detection of motion, right?
Yeah, actually, the DALAM meeting that Pete had organized in Berlin 9 years ago was very eye-opening. So I was a clinician among many scientists. It was humbling, but very humbling. But clearly, these questions came up about how to get more efficient opsins, and these were among the many questions. This format with so much brainstorming was very, very intriguing, and certainly the idea of using the on pathway and the off pathway with selective proteins and selective vectors is certainly one of the ways to go. But the good thing is now we know it can work, but it can work much better, certainly in the future.
What do you think for the development of the Googles? Because I think this is also important, maybe to include machine learning inside. Is it possible, do you think?
Yes, so currently what is nice is that existing cameras have a lot of processing inside. For example, I used to say that we try to transform blind patient into low vision patient, which is not very ambitious, but for them it's very ambitious. But low vision devices exist already that have a lot of vision processing. So we know about the goggle glass that never worked, but we also know that there is a technology called ORCAM, where the system is telling the patient about, patients don't like it so much, but it tells them about the object they are seeing. And there are other technologies that are used, for example, in age-related macular degeneration to improve edge selection. What we like with these cameras is that they don't benefit from this system, but they are working real time and they are detecting any change. So it does reduce the amount of information that the retina and the brain have to process by many log units, because if you imagine that when we are seeing a lot of things that are not moving, that are not informative all the time, and the brain has to decide what is important and not important. What is moving is usually what is important. So we believe this is better, and the patient tried the frame system with fixed images versus this system, and they preferred the system with the vibration, probably because of the enhancement. But I agree that more machine learning and more enhancement of signal would be very helpful.
So in conjunction with the lectures we heard this morning, another probably more obvious application would be to complement the defective gene by a correct gene.
Yeah. So this is my daily life. So this is what we do all the time.
Yeah.
Currently, as you know, there is one drug that was approved a few years ago in the US and in Europe, where we are correcting the gene defect, in that case in the supportive cells, the retinal pigment epithelium. And my department, we treated 20 kids with that, and it works. It helps them not to have a normal life, but it changes their lives. We have now many trials ongoing. Some are successful, some are not successful. But the key question, I tried to allude to that in my talk, is that you need viable photoreceptors, and you need photoreceptors that can be transduced efficiently. So first, they have to be alive, and second, they have to be in a stage where are not already in the phase of degeneration, where you are going to correct the gene defect, but it's too late to correct that. And so this is something that we are working on, but many are working on, but there is a time window for that. The other issue is that it's a gene-specific approach. Even the CRISPR-Cas is actually a mutation-specific approach, which means that within the field of orphan disease, you have sub-orphan populations, and we see now that a few diseases are being tackled by big companies, but many diseases, there are not even enough patients to be targeted with these approaches, and this is a big economic problem that we have to face in the future, how to target these very rare genes, these very rare mutations. So this is why gene-independent approaches have a beauty.
Yeah, but repair is something different from complementation.
Of course.
Yeah, so complementation probably is easier than to repair, from an ethical perspective, probably.
Maybe, but repair, I mean, the gene editing is repair. The gene addition is just it's complementing. It's working. It's working, and it's working. We have an issue about overexpressing, which might be a problem in the future, but still, this is something which is currently working. But when you talk about dominant disease, rhodopsin mutation, for example, still a few approaches are in development about inhibiting the wrong gene and overexpressing the other one, but the ratio and the amount of protein, as you know, overexpression of proteins can be toxic to the cells too. So these are some of the issues that we are facing. But this is a field in full development, and actually there is something which few people talk about is the stoichiometry of proteins, because it's not like a plug and play. You are just going to bring back the protein, and the cell was just waiting for this protein to come back, and everything is going to adjust to it. The cell has been dysfunctional for a long time with this missing protein, inducing a lot of cascade of events. Are you going to reverse all of that? We don't know yet.
Maybe to get a vision, do you think that it's possible with this approach to have to restore also color vision? In principle?
Yeah, so in principle, if you target the cone photoreceptors, you could have at least dual vectors, and we have an approach in development currently on that. So in theory, it would be possible. It could also enhance the contrast sensitivity, not just the color vision, but it could also help to improve the contrast. But this is early stage currently. Yeah.
Maybe the last question. One more question.
One more thing? Yes, please.
Yes, I have one more question, because about 5-6 years ago, everyone expected more or less that the channelrhodopsin would disappear because to use cone pigments would be more efficient because you have this amplification.
Yeah.
Is this still an option?
Yeah, actually, big pharmas believe that. So they invested a significant amount of money into that. We haven't yet seen anything come out of it, but it's the early stage, so it makes some sense. I'm still concerned that you are not going to restore the adaptation and all the cycle that does exist in the classical phototransduction. So I'm concerned about patient adjusting to many levels of light. I don't see this happening, but we'll see. There is one company that has press releases, no publication, but a lot of press release about color vision and everything. So waiting to see the paper. I've not reviewed it, so I don't even know if it was submitted, but waiting for that to see whether this is real or not real.
Okay. But the expectation of the scientists is smaller than the expectation of big pharma, right?
Oh, yes. Well, actually, the expectation of big pharma is more the market, that's all. Yeah.
Okay. Thank you very much.
Thanks to the three of you. I could listen for ages. So, but now welcome Oliver to present his slides, and we go on with a different angle on that one.
Yeah, so hello everyone. Thanks for having me here. Congratulations, José, to this very inspiring speech, and it's definitely a difficult task to follow you and this very scientific discussion here. Sight-threatening and blinding diseases are devastating diseases. Hence, these diseases deserve public attention. At Charité, we are in the Department of Ophthalmology, a group of experienced PIs looking at primarily retinal diseases from various angles. The findings that we have, we compile that and put that together in integrative systems medicine model. Our belief is that systemic factors, just as genetics or the metabolic state, determine the cause of key disease processes, such as inflammation or angiogenesis, which play a key role in important eye diseases. And by this means, systemic factors influence eye disease. At the same time, we understand the eye as a window to the body, because the eye is the only place where we can visualize neuronal tissue and vasculature in the living human subject. And you can imagine that this window function of the eye opens exciting avenues for interdisciplinary research. The macula is the center of the retina, and it is responsible for sharp vision. So if you look at this slide and read it, you need your macula. These are two scenes, everyday scenes here in Berlin, through the eyes of a healthy individual. And this is through the eyes of someone with macular disease. You can appreciate that this is not only coming along with some discomfort, but that this also causes peril, such as the risk of fall. And it's very common, this condition. So we have, alone in Germany, millions of people are affected. VEGF inhibition was a game changer in that field. With VEGF inhibition in neovascular AMD, we can preserve vision in 95% of subjects, and about 1/3 we can improve vision. This is remarkable, since before that, blindness was inevitable. The drugs need to be injected into the eye. The treatment reality of a patient, however, looks like this. So it's injections over injections, often every month. So one important research topic in ophthalmology is how to reduce these injections. And in my former life at Bayer, I had the privilege to lead a team which got the first compound approved which produced similar outcomes with every other month dosing than with monthly dosing. So with that compound, it looked more like this. So there was or is more time in between the injections for the subjects. However, we are still not there, and this journey continues. We looked at replacing injections by eye drops. However, with that project, we failed. And this raised important questions and showed us where the demand is. We need to revise the disease understanding, we need new preclinical models, and we need to establish more predictivity along the development chain. That's what actually something which brought me then transitioning into Charité, into academia after several years in pharma. We have projects underway in our group like the BioMax study. This is a system medicine study in neovascular AMD, where we've identified certain angiogenic factors that are systemically present and determine the need for injections in patients with neovascular AMDs. So this system medicine project in this disease shows, yes, there are systemic factors influencing the course of disease, and we're heading in the right direction. I personally believe that the vivid research landscape that we have here in Berlin is the perfect place to be, to do this kind of research and hopefully prepare the grounds for the next big breakthrough in ophthalmology. Preventing blindness and preserving vision is intuitively linked with quality of life, or in other words, research that prevents blindness makes a difference. And this is where we all as translational scientists are striving for. Thank you very much.
Thank you, Oliver. It would be great if you came back on stage, and I'm sure you have some questions to ask for José.
Sure. So I'm very interested. Of course, we're talking mainly about monogenetic diseases, which are a fraction of the retinal diseases. How do you see genetics, gene therapy, gene transfer in these complex diseases such as macular degeneration, which fill up the outpatient departments of the eye clinics? Do you see something on the horizon?
So, as you know, probably better than myself, a couple of companies are developing gene therapy to deliver. So it's not curing the disease, just delivering therapeutic genes, for example, the anti-VEGF proteins to the retina. So this is ongoing, and currently the odds are this may work, but you may not want to have a permanent inhibition of something for life, because the disease may not need that forever. And with gene therapy, there's an irreversible part to it that is sort of a concern. But still, it could reduce significantly the number of injections, so it could improve the compliance. What we know now in real life is that the 95% and 30% you have on your slide are from the clinical trials. But in real life, many patients don't come back as often as they should. Many ophthalmologists don't see the patient as often as they should. And there are a lot of patients that are not really benefiting from the optimal status that were obtained in the clinical trials. So this could be an approach, but it's not a cure. So there are other approaches currently targeting the inflammatory pathway that are using also gene therapy. And at least one of them was announced as positive, but still questionable currently. What may happen is that some of the approaches that I discussed, the one on neuroprotection that I didn't talk about, but which was my real project for 25 years and is getting into the clinic next year, this approach could apply to age-related macular degeneration because it's protecting the remaining photoreceptors, so it could have an application to this disease. The issue with age-related macular degeneration is twofold. One, patients still have peripheral vision, so you don't want to do anything that could harm the peripheral vision, and you want to combine the two. And the other one is that there is no good animal model because there is no animal with a macula. So everything we do, there is a big leap between the preclinical work and the clinical work. This is why anything we are learning on genetic disease can be very informative, because once we know that it is safe, then it gives you the ground to get into a clinical trial, as long as there is some biological rationale to do that. So I think it's in the horizon. Curing the disease by gene therapy, I don't think we can, because it's a very multifactorial process and probably only one of them. But trying to deliver some agents or trying to reactivate or protect the remaining cells is in the horizon.
Provocative question, maybe. Sure. Do we know enough about the causes of these diseases?
So we thought we knew, actually, in 2005. So the VEGF was one of the cascade pathway, but in 2005, so many papers came out on the complement factor and the inflammatory cascade, so everyone thought that, okay, we know what is the cause of the disease because it's genetic and it does explain a lot of things. Now we know it's far more complex. Even the function of a complement is not the one we thought about. So, but everything in science is like that. We think we know, and we just learning more by mistake, and we heard a very good talk about learning from something which was unpredicted initially. So I think we don't know enough, but we know at least that this is one of the targets that could be approached.
My perspective is also having experience with targets beyond VEGF, that we were very lucky with the VEGF that this was working. That it is working and that it was working that well.
Yeah, it's a miracle.
My personal opinion is that we need to expand the target space in macular degeneration.
Yeah, absolutely.
And maybe we open also up to the audience, if Christiane agrees to.
I think that's fine.
to that, yeah. Whether there are questions to José or to the other panelists.
And there was one remark from the technicians. Please wait until Shamram is there with the micro, so that the people who watch this via streaming can hear. Great.
You told us you worked at Big Pharma, and then you returned to basic science. Why? Was the Big Pharma approach unsuccessful? And what was the approach? What did they want to
I'm not calling it return. I would not say return. I would say I continued to academia, so it was not a going back for me. The reason is that I'm an ophthalmologist and I'm very much dedicated to ophthalmology. So in Big Pharma, of course, you could also then transition from ophthalmology into oncology or into cardiovascular and then do a career in marketing also. But I'm an ophthalmologist. I'm a research-driven ophthalmologist. And I realized exactly what I tried to imply with my questions to say that the basics are not there to repeat or to continue successes like the introduction of VEGF inhibition. So we need to understand these patients better, the disease better. We maybe even need to re-stratify these diseases. We call this all age-related macular degeneration, but in reality it's maybe more than this. And this is, in my view, clearly the domain of academia to do this type of research. Of course, theoretically, Big Pharma could also do it, but this is a product-independent research. This is an open research which also needs to be accessible, not protected behind patents. And therefore, I believe that this is a real strength of academia to do this kind of research, and this actually made me attracted me and made me then make this move.
I'll take this one, if I may. José, extremely impressive and very inspiring. Also, your enthusiasm after all of those years is really incendiary. And if I may, a technical question. So you showed us 2 movies in your presentation, and in one you showed how this camera works, and this camera apparently just detects differential light. But then in your Pioneer trial, you made the patient detect a static object where there shouldn't be any differential light. So how do the 2 things go together?
Yeah, so, well, first we had to start somewhere. And so we started with an object that the patient had to detect. So if we added complexity with a moving object while the patient, what you may have realized that one of the key challenges we still have, is that we are only transfecting a small area of the retina, and the camera has to be fully aligned to this area. A lot of the training is the alignment. So if we add to that movement, it becomes an issue. But the patient himself is moving. If you look at the movie, the patient is moving, which is part of the enhancement probably of the signal by the movement of the head, of the eyes. Because the visual field is not large enough, probably he has to move the head currently. And this is the experience I have from artificial retina. The patient also had to move because it does sensitize the ganglion cells better, but it does compensate from one of the issues we have, which we are activating all the pathways at the same time. So we cannot fully translate the science into a clinical, but we have to start somewhere. And the main issue is really the fixation.
But did you then use a different camera? Because this camera actually shouldn't see that object, isn't it? So I mean, I get the patient part, but I was confused because this camera shouldn't see the object because there was no differential.
Any small movement is creating more contrast on the object. So any small movement that the patient is doing is enhancing actually the signal. And the patient actually, if you heard, he was mentioning vibration. And we tried a static camera. It turns out that for the artificial retina, so we have an upcoming paper on a subretinal artificial retina, and the patient didn't like this type of camera. They preferred the static camera. But for this trial, at least as of today, but we are still learning from the patient, they prefer this vibration because it does enhance in some ways the signal. But it's very pragmatic. It's not rocket science, that's what you heard this morning. Rocket science is preclinical, but the clinical is really pragmatic.
Something? Yeah.
So you told us a lot about gene therapy approaches. Could you tell a couple of words about cell therapy approaches? I mean, in Japan and also I think in London, Project to Cure Blindness, they use, at least for AMD, already retinal pigment epithelial cell plasters to bring into the back of the eyes. How far are we with photoreceptors? I mean, you can make photoreceptors, I guess, also from iPS cells. So is there any idea to bring those in already into the eye? And then maybe something on AMD, how you see this developing with the cell therapy and maybe combining it with gene therapy as well.
Okay, so it's 5 questions. I'll try to. Yes. So cell therapies are ongoing, and they are promising for the pigment epithelium. For photoreceptors, the issue of reconnection is still not fully solved, and there was a couple of papers in big journals where it turns out that it was probably material transfer between the transplanted cells and the host cells. So this is still controversial. For RPE, there are many approaches. Actually, my group, we published in Science Translational Medicine about using a substrate and RPE cells on that. The London group is doing that. The Humayun group in California is doing that. So all of this is in clinical trials currently, and the level of vision restoration is very variable. Actually, our approach was to start that again in retinal degenerations because we understand better what we do, and these patients are blind, so the risk is limited as compared to any effect. Like you have to remember that for all these therapies, except the Japan approach with iPS cells, these patients have to be put under immunosuppressants, and they are elderly patients, so this is some of the concern. So the iPS, as you know, the Japan study with Masayo, they did one patient, then they had to put a hold because of the process of transformation of the cells. They found that there was an oncogene that could be activated, so this is still a bit controversial. Until we have a library of iPS cells from the population that are healthy and considered as safe, this is still going to be an issue because taking the cells from the patient and transforming them, you always have the risk of some potential oncogene that could be activated in the process. But this is one. The challenge is really the reconnection. So we published 2 years ago in Nature Communication an approach where we use progenitors of cone photoreceptors. Actually, Marius Hader in Germany and Stefan Erlis were part of that work. And we used optogenetics to express the optogene in the stem cells that were precursors of cone cells. So we did organoids from iPS cells, so organoids 3D. We did triage to just get the photoreceptors, transfected them with optogenetics, and put them back in the mouse eye. And we could show a reconnection, including the activity at the level of ganglion cells and behavioral responses. So this is something which we are trying to translate into human trials. But as you can see from the steps, it's not going to be a simple process, but certainly one of the promises for the future. Yeah, the beauty of optogenetics is that you are using the cells that the patient already has. You don't have to bring foreign cells, and so you're reducing a bit the risk of the technology, but still there.
Maybe also from a more clinical perspective, going very much back in time, 20 years, what we did back then is that in patients with AMD, with macular degeneration, we detached the entire retina and then turned it a little bit so that the macula of the retina came to be on RPE, which was healthy. Yeah? So, and that worked in principle. Patients saw better, but what happened in many patients, besides all the surgical complications, but what happened then in the patients is they saw better for a while, and then after a short period of time, the retinal pigment epithelium under the macula was degenerated again. Yeah? So, and this brings me back to the theme. We do not need enough. We do not know enough about these diseases. We need to know more. Of course, it's an quite easy one to take a cell and inject it somewhere. Of course, also difficult, but it's in the end, from a surgical perspective, it's an easy thing. But are we doing the right thing here? And this is where I strongly believe we need to know more about these diseases. And maybe other short aspect to it is also we should not forget. Macular degeneration is extremely frequent. I spoke about millions of patients. Not all of these millions would qualify for cell therapy, but it's still hundreds of thousands. Yeah? So we need also a therapy which is practical and which can be offered to at least a majority of these patients.
So it's 1 o'clock. I think it was a beautiful hour. It was a pleasure looking at the four of you and the love and passion you have for your work from the different angles, and not only love and passion for your work, but love and passion for this panel too, because we didn't rehearse that, and I think it worked out perfectly well. So we meet here in 1 hour's time. Lunch is downstairs, I guess. So we continue with diabetes, which I think is a bit mean after lunch. But please come back and enjoy your food.
Thank you. Thank you so much.
Okay, everybody, welcome back after lunch. I think we should continue. And now we're talking, as promised, about the breaking the wall of regenerative approaches to treating diabetes. And it's a lifelong condition. And the cells in the pancreas are either not working at all, or they are working a little bit, not enough, to produce insulin. So patients have a high blood sugar level. And so we welcome the winner, one of the winners of the Breaking Walls this year. So it's Professor Heiko Lickert. And he is director of the Institute of Diabetes and Regeneration Research at Helmholtz Center Munich, and chair of beta cell biology at the TU in Munich. He's a developmental and stem cell biologist and has devoted his career to understanding how the pancreas and the insulin-producing beta cells are formed and how we can use this knowledge to find a cure for diabetes. So findings from his studies serve as a basis to either regenerate patients' beta cells or replace them. So a newfound receptor plays an important role in this, and Heiko will elaborate on this as he's one of the winners at the Falling Walls today. And then we do have as a matching partner from the Charité, Professor Joachim Schwanger. He is the medical director of the Department of Endocrinology and Metabolic Diseases here at the Charité. He saw patients this morning, so this is why he wasn't here. And he's a diabetes expert. So recent research includes the relation between obesity, fatty diet, diabetes, and signaling in the brain, and the role of the microbiome in diabetes. So Joachim will give us an overview over the field and the upcoming therapies for the metabolic disease. The two of them didn't know each other before, but they got on really well. So I'm looking forward to this.
Yes, thank you very much for the kind introduction, and also would like to thank the jury, obviously, for selecting this project for this year's winners. And I'm really happy to be in Berlin, and I think it's only Berlin which can have such progressive meetings like this. I'm really enjoying it. So what I want to talk to you today about is how we think or how we want to break the walls to regenerative therapies. And it's just now after you had your lunch. It is when the beta cells really have to work very hard to produce insulin. And if that does not happen, then you become a diabetic patient. And so we just heard there's two ways how you probably could treat the disease. It's once by replacing the beta cells from stem cells or trying to protect the beta cells in pre-diabetic patients or regenerate them. And that's what I want to talk to you today about. Now, but what I really have to start with is we're celebrating also this year the 100 years anniversary of discovery of insulin. And it was already in 1923 when Frederick Banting mentioned at his Nobel Prize lecture that insulin is a therapy but not a cure. And that has unfortunately not changed until today, 100 years later, and explains why we have 470 million people suffering from diabetes worldwide. And that number is going to be increased in the next 25 years to 700 million people. Diabetes is not only a global problem, but it also causes a lot of deaths. And it actually causes more deaths than corona does. And we don't have any vaccination for diabetes. And that makes obviously also the healthcare costs exploding. From the Robert Koch Institute, we know that 20% of the mortality in Germany is due to diabetes. The problem is that we do not have any curative approaches. So all the medicines out there are symptomatic treatments of the disease. And over years, the patients develop devastating micro and macrovascular complications, which you can see here. And that is why there is a huge pressure on us as researchers, but also on investors and pharmacy to really invent or bring new drugs to the market, which actually can be disease modifying, which can treat the root cause of diabetes. Now, you probably know that we distinguish two forms of diabetes. Type 1 diabetes is an autoimmune disease. Usually it manifests early in life. And it affects 5% of the whole diabetic patients worldwide. The reason here is that beta cells in the pancreas get destroyed by autoimmunity and leads to early onset of disease. The bigger problem is type 2 diabetes. 95% of the people suffer from type 2. And so they are insulin resistant. Obesity is a risk factor for type 2 diabetes. And there, in the beginning, the beta cells try to compensate for the metabolic need, but then fail. And that's when diabetic patients become diabetic. Pre-diabetic patients become diabetic. Now it is really when the beta cells in the pancreas, which measure glucose and secrete insulin postprandially after a meal, when these cells give up, when these cells do not function anymore due to different stressors, which I just explained, autoimmunity or glucolipotoxicity, when they fail, they become dysfunctional, they de-differentiate, and eventually they die. So it is clear that if we could protect these cells from failure by protection or regeneration, we would fight the root cause of the disease and could develop disruptive therapies. And that's exactly what we're doing in my institute. Now, a few years back, we discovered that beta cells are not all the same. So beta cells come in different flavors. We have beta cells which are more proliferative, and we have beta cells which are more functionally mature in terms of glucose sensing and insulin secreting. And we really think that this understanding, this heterogeneity is key to basically implement new methods or new drugs to maintain beta cell function. Now, this is an islet of Langerhans. It's a cartoon. So this is a very small mini organ in your pancreas. It makes approximately 2% of the pancreas. And here in this islet of Langerhans, the beta cells are wrapped around blood vessels, and they sense glucose and secrete the hormone insulin into the bloodstream. And the majority of beta cells are really doing this job. They're there for exactly this function. But there's a minority of cells, approximately 10% of these cells, they have so to say, compensatory potential, and when there is metabolic need, for instance, in obesity or pregnancy or insulin resistance, it's these cells which can proliferate or which can basically mature into functional cells. And so the big question obviously is, can we harness this heterogeneity we have identified to protect or regenerate beta cells? And I will show you an example which let us believe that this is the case. But before I do so, I have to introduce how we think or how the paths of regeneration can be triggered. And I will show you more about embryonic development, but it's the multipotent pancreatic progenitors which give rise to all the different cell types in the pancreas. To the endocrine portion, the 2% of islets of Langerhans, for instance, the glucagon-producing alpha cells or the insulin-producing beta cells. And so these multipotent progenitors in a process of neogenesis form these islets of Langerhans. And it's clear if we want to trigger regeneration, we have to understand how beta cells are born and how they are formed. There's still a debate out there if there is something like adult stem cells in the pancreas. But it's clear that the pancreas is not something like the gut or the blood or the skin, where there are highly proliferative adult stem cells which replenish and turn over the organs in, like for the gut, in a week's time. So the pancreas is not such a niche for adult stem cell. But it's possible that there is a very minor turnover. And in mice, actually, we know that these stem cells do not exist, but we do not want to exclude that a human being which lives much longer may have these facultative stem cells. The main mode how beta cells replenish and regenerate when there is need is by self-replication. So one beta cell makes two new beta cells. When you are, for instance, insulin resistant or obese, and understanding the mechanisms how cells self-replicate, beta cells self-replicate, is obviously a way to trigger regeneration in the pancreas. There's experimental evidence that cells which are closely lineage related, for instance, the alpha cells, can be transdifferentiated into beta cells by just expressing transcription factors, beta cell-specific transcription factors. Now, if this is clinically translatable, is still questionable because you would need gene therapy or mRNA therapeutics to do so, and it needs to be very specific. And having drugs like insulin or metformin on the market makes it unlikely that gene therapy will be a therapeutic way of basically treating diabetic patients. Now, last but not least, we are very interested in a concept which is called dedifferentiation. So upon stress, cells lose their identity and they go back in developmental history. We call this dedifferentiation. The good thing is they're hiding from the stress, so they're still there. The bad thing, obviously, is that they're not functioning anymore. So they do not produce insulin and do not sense glucose anymore. But if we could convince these dedifferentiated cells to come back into function, there would be also a way of regenerating pancreatic beta cell mass. And that's exactly what we have published last year. So in this experiment, what we did is we ablated chemically 80% of the beta cells in the pancreas. This is not sufficient enough to sustain blood glucose levels. So these mice become hyperglycemic, so they become diabetic. And then we looked at the fate of these beta cells in a diabetic hyperglycemic environment. And what we saw is exactly what the scheme was showing to you, that the beta cells dedifferentiate. They lose their functional identity. They upregulate stress programs, which are actually entry sites for us to target these cells. And then we used single-cell technology to look actually at the fate of dedifferentiating and redifferentiating beta cells by doing pharmacological intervention. So we gave these mice over 100 days, we gave these mice the standard care drugs like insulin or like GLP-1, which is a gut hormone, which is also on the drug market. And we also hooked up this gut hormone, because the GLP-1 receptor is sitting on these beta cells, to specifically target estrogen into beta cells. And why did we do this? Estrogen is known to protect postmenopausal women from diabetes, but it also has side effects. So we shuttled in estrogen into beta cells. And by looking just at beta cells, at healthy beta cells using single-cell RNA sequencing, or dedifferentiated beta cells on the other side of the spectrum, you see here in color all the different treatments we had. And when you're looking here at the red and pinkish color, this was exactly what insulin and the combination insulin, GLP-1, estrogen did. It brought back dedifferentiated beta cells into something almost mature. And that was enough to cure the mice from diabetes. So these kind of approaches show you the paths and the mechanisms of de- and redifferentiation, but also identify many novel targets which you can interfere with to protect or regenerate beta cell mass. But to understand regeneration, I said it before, you have to understand the generation of beta cells. And that happens during development. Now, I said that the pancreas is an exocrine and an endocrine gland. It generates enzymes, which are secreted into the duodenum for digestion. But it also generates the endocrine part during embryonic development, where pancreatic progenitors give rise to acinar cells, which produce the enzymes, ductal cells, which train the enzymes into the duodenum, and endocrine cells, which are important for blood glucose regulation. And it's essential to understand the signals and factors which regulate this process, that, for instance, we can generate beta cells from stem cells in a culture dish for cell replacement therapy, but also for disease modeling or drug screening. So we can translate this developmental regulatory logic, but also developmental biology, understanding developmental biology has helped to identify new disease genes. And one example I want to bring you today. So to understand development, you really have to understand it during time. And here we knocked in reporters under transcription factor control to look at this process of endocrine and islet development. And these orange cells you see forming in a pancreatic explant culture of mice. So these cells, they generate the islets of Langerhans. And then combining this technology with single-cell omics across evolution in mouse, pig, and human can now give you very precise maps, road maps of what happens during this differentiation process. And so this is a lineage trajectory of multipotent progenitors here in a porcine model where you basically see very nice how these progenitors differentiate into pre-acinar, pre-ductal cells, but also into endocrine cells and then in beta cells and alpha cells. And these maps we can only generate nowadays where we have the single-cell omics technologies. And by this kind of expeditions, we identify novel genes. So this is a gene which didn't have a name when we discovered it, but it caught quickly our attention. Because in a mouse pancreas is specifically expressed when beta cells are born and insulin is made, and it had domain signatures of the insulin IGF signaling pathway, which regulates growth, proliferation, and metabolism. And so to cut a long story short, what this inceptor does, 100 years after discovery of insulin and 50 years after the discovery of the insulin receptor, we identified this insulin inhibitory receptor, which binds to the insulin receptor in the activated form on the plasma membrane. And what it does, it basically internalizes this activated complex and it switches insulin signaling off, specifically in beta cells. Now, a lot of work in mouse and human model systems has then revealed that the knockout phenotype of this inceptor knockout obviously shows increased insulin and mTOR signaling. And that is important for the differentiation, the survival, but also the function of beta cells. So beta cells do not only make insulin, but they also require this. Now, to target this molecule, we have developed antibodies which bind to the extracellular domain of the inceptor. And by steric hindrance, it doesn't allow it to come together with the insulin receptor. And that is how we can sensitize insulin signaling in mouse and human beta cells. And to show that these antibodies really work in diabetic mouse models. We have basically used here a very aggressive type 2 diabetes models where beta cells decompensate. So this is an isotype control. We injected antibodies over several weeks. Every 2 weeks, a dose of antibodies. And in the control, you see that the islets of Langerhans really decompensate. The beta cells get lost. And with the anti-inceptor antibodies, we can actually preserve beta cell mass as well as beta cell function. And we're currently humanizing these antibodies to bring this to clinical trials. Now, what I've told to you today is that Inceptor is a very interesting target which counteracts insulin signaling in the pancreas, and we think it is important at the source of insulin that you have protective mechanisms that the cells do not show or see autoactivation. It does so by clathrin-mediated endocytosis of the activated insulin receptor complex, blocking the inceptor by genetic means, I haven't shown to you this, or by pharmacological means, can increase beta cell proliferation, protection, survival, as well as function. And so that's why this molecule, by studying developmental biology, was a very curious finding. And we hope that we can use such information to protect and regenerate beta cells in human in the future. So I would like to thank you for your attention, but especially I would like to thank all the people working hard in the lab to make these discoveries possible, and also the funding agencies. Thanks very much.
Thank you, Heiko. And now I think Joachim will take over.
Well, thanks very much, Heiko. I think you took the... right. Thanks very much for this wonderful talk, and thanks to the organizers for the opportunity to frame what you told us and celebrate our prize winner, of course. I will try to give you a short overview. The topic of the falling wall prize-winning work was, at least that was what I read, is the treatment of diabetes. That is very interesting, and treatment is one goal. I think what we really want to do is to cure diabetes. That is the holy grail. In terms of treatment, we have made great progresses in the recent years, and you started with the Benting discovery 100 years ago of insulin, and meanwhile, just to name one example, we have so-called smart insulins. So smart insulins are insulins that are interlinked by chemical molecules that are glucose sensitive. So the release of insulin from the subcutaneous tissue after injection occurs in a glucose sensitive way, and what we want to get is basically to avoid hypoglycemia. So as soon as the glucose is low, you don't want any further release of insulin. So that is shown schematically here. These are the smart insulin which are not further released after you have a normal blood glucose, and that works. This is shown here in a clinical trial. So as soon as the glucose is down to something like 100 milligram per deciliter, you see that the release is ending, and you get a further increase as soon as you again increase the glucose, and the rate of hypoglycemia is lowered dramatically. Just one example to show treatment, we have really made great progress, and I think Benting would be excited to see those data. We have other breakthrough findings. We have drugs, for example, which affect the excretion of glucose via the kidney. I think some years ago, decade ago, nobody would have expected that the metabolism of glucose in the kidney may be a major druggable and treatment approach. Apparently it is. And we have a number of really great and large studies showing it's not only improving the glucose, but it's also improving cardiovascular endpoints. And this is not only the case in diabetes patients, but it also works, for example, in patients with heart failure and kidney impairment or kidney failure. So that is another breakthrough, which is absolutely impressive. A number of other treatment breakthroughs, I think, could be named here, but as I said, I think the holy grail really is to cure diabetes. Is it possible to cure diabetes? Yes, we know that. We know from bariatric surgery, so these are surgery approaches which result in a dramatic and sustained reduction of body weight. in individuals who are morbidly obese, so BMI above 40 or above 35 and having diabetes or other complications. And we know from these data that we get a sustained and long-term reduction of body weight. And these people have a remission of diabetes, which is very high and very impressive. And that's what we would think is a kind of cure of diabetes. So theoretically, yes, and practically, it's possible. But apparently, bariatric surgery is not the ultimate goal, which we would like to see in every patient. So we need other approaches. And if you look into the pathophysiology, and this is a very old figure, which I like very much because it shows the interaction between insulin sensitivity and insulin secretion very nicely. It's a figure by Christian Weyer from 1999, where he demonstrated it's always the interconnection between insulin sensitivity and insulin secretion, which is important for the long-term glucose control. So this is insulin sensitivity, or an estimate of insulin sensitivity. This is an estimate of insulin secretion. We all have more or less, at any times, if we have an infection, for example, we have a worsening of insulin sensitivity. We develop insulin resistance. So we move here on the curve to the left side. And we usually cover that by increasing insulin secretion. So no problem, the glucose control remains the same. Just if both comes together, so we have a worsening of insulin sensitivity and we cannot cope by increasing the insulin secretion, then we develop impaired glucose tolerance or diabetes. So at the beginning, I would say in the last 30 years, many studies worked on the insulin sensitivity and focused on the organs which are believed to be central for the development of systemic insulin sensitivity or insulin resistance, so the liver, the muscle, the fat. And to be honest, the success of drugs which based on these findings was very limited. One made it, for example, into the clinic that were the so-called PPAR gamma agonists. PPAR gamma is a transcription factor which is important for the differentiation of fat cells. And there was a very good rationale to believe that this improves insulin sensitivity, which it does. But at the end, when we looked into the large trials, the patients had an improvement of insulin sensitivity, but unfortunately the cardiovascular risk was even increased for some of those drugs. So this was unfortunately not the final answer. But I told you bariatric surgery is a good example. So the question may be, if you want to prevent diabetes, why do not prevent obesity, which is the major risk factor? And I think at that field, it looks a bit different. Since the finding of leptin, which is one of the major regulators of energy homeostasis, we have identified a complex and redundant neuroendocrine network which is responsible for the very tight and very close regulation of the energy homeostasis. And we learned that not one single factor can be responsible or can be used to treat obesity, but we need to target more than one of those signaling cascade. So here, for example, is a study from Matthias Schöp from Munich, and he looked into a so-called triple agonist, and that targets not only GLP-1, which is a gut hormone regulating energy homeostasis, but also GIP, which is also a gut hormone, and glucagon. So 3 hormones which we know are responsible for the regulation of energy homeostasis. And what we see is that the effects of the single signaling cascades is additive. So if we target more than one signaling molecule, we get an additive effect, and these are, for example, very impressive data with a 20% weight reduction with these drugs and the dual, but also the tree agonists are meanwhile going into clinical trials, and I would expect that they may at some points come into the clinical practice. For example, GLP-1, which is one of those signaling molecules, is already in the practice. So GLP-1 analogs have been approved for the treatment of obesity. It's just not sufficient what we see in terms of an effect size, but clearly it shows that the mechanism works. There are many other factors which may play a role, and I just want to show what we work on. We ask the question why people regain weight after they initially lost weight, and we have recently shown that, for example, the microbiome, which is one of the new kids on the block, is dramatically changed if a person is reducing its body weight. And the interesting issue is this contributes if you transplant this changed microbiome to gnotobiotic mice. We see a totally different, a completely different development of body weight compared to those mice who receive the microbiome before the body weight. So that shows that other factors, not only what we have seen, like the gut, but also the adipose tissue, but also the pancreas, but also things like the microbiome signal to the brain, and may be important if we finally want to get druggable targets. So coming back to our initial figure, I think what Heiko, and what is so impressive in Heiko's work is really that he interlinks these two issues. So the insulin sensitivity and the beta cell. What he has shown is that the beta cell does have insulin receptors itself, and I think it's a major finding how this signaling of the insulin receptor on the beta cell is regulated and what that means to the glucose control. So fusing these two ideas of insulin sensitivity and the beta cell work, I think, is one of the major findings of Heiko's work. And I think it's very interesting to learn, and it will be interesting to learn what the future brings. I mean, Heiko has shown us two possible ways. One is the redifferentiation of beta cells with the example of a target or a beta cell specific targeting by the estrogen coupled GLP-1. I think that is an excellent idea and may prove to be effective. The other idea certainly, and many people are working on that, is the development of islet organoids, in which way soever, which may also work. And we have very recent data, and that is my last slide, from a company, Vertex. They are working on allogenic development of allogenic islet organoids, and they have started a clinical trial and have just released by a press release the result of a first patient, a type 1 diabetes patient, and they show that they can increase the C-peptide production by these islet organoids substantially, and can reduce the requirement of insulin treatment and the dose of insulin substantially. So certainly there is a way or a path how islet organoids may be used, and it will be very interesting what the next steps in terms of beta cell biology will be. In any case, I think the work of Heiko is very important because we need to understand the pathophysiology before we can finally develop a reasonable treatment. So with that, I would like to end. I would like to call Heiko back. I heard we have or should do some kind of discussion. And my first question, Heiko, would be, you probably expected already, what would you think? I mean, you have shown some opportunities, transdifferentiation, redifferentiation, islet organoids. What would you think, what is the best? Where should we put the money to?
Best you do not put all the eggs in one basket. So that's. So I think you mentioned it. It's very intriguing that SEMA Therapeutics and Vertex has now reported on this first clinical trial of beta cells from stem cells and had really very nice outcomes, effectivity. They showed it. The cell replacement approach, as you can imagine, also requires, first of all, that you have cell material, and it's not easy to generate beta cells from stem cells. It's a lengthy procedure. It's very it's very expensive, and you have to protect, when you have generated the cells and transplanted the cells into the patient, you have to protect them from rejection. And on top of it, usually that approach is more suited for the type 1 diabetic patients, which basically do not have beta cells in their pancreas. But they also have on top of that the autoimmunity. So if you bring beta cells back into this patient, they will be seen by the immune system again and attacked again. So we'll have to find ways of hiding these cells from the immune system. But then these cells really need to be very safe. So currently there's still a lot of hurdles to overcome. The other approach we were talking about, beta cell protection and regeneration, I think is probably applicable to a much wider patient population, because you can basically medically treat these patients with pharmaceutics or pharmacology. But also here, I've talked about beta cell proliferation as one of the targets. And it is clear that in the pancreas, if you want to cells proliferate, make them proliferate, it has to be very specific to the beta cells. You should not get any exocrine cells proliferating, because you don't want to induce any tumors. And that boils then down to ways of getting selective targeting of cells doable. And I think this is a challenge for all the diseases. Get basically the treatments more selective in a way that you, also in cancer treatment, that you only treat the cancer cells and not deliver your drugs systemically.
What I feel, I mean, I would like to come back to the islet organoids, which I think is very attractive, because you can modify the cells before you inject them or differentiate them. What are the major hurdles currently? I understand what we get are organoids which are not so glucose sensitive in a way. I wondered about the Vertex trial to be open. I mean, they injected that just in the portal vein and then into the liver. I think nobody knows what that means on the long-term safety, but perhaps you can comment on that as well. But what are the major hurdles to overcome in terms of developing functionally active islet organoids?
Yeah, I mean, you have seen an example from development. I've shown you pig development takes 110 days. Human development takes 40 weeks. And so we are generating cells in culture dishes in 3 weeks. So I think you cannot expect that in 3 weeks these cells are perfectly functional and mature. But I have to say the field came a long way. These cells, which we currently can produce in a culture dish, are already quite good. They can respond to glucose and they can make insulin, but they're not perfectly glucose responsive. So there's still a lot of hurdles to overcome here to make better cells. And I think the huge, what we haven't talked about, you know, the huge advantage of having stem cell derived beta cells also allows you to recapitulate disease, which in the human body probably takes place over many years. And we should say that the pathogenesis, you know better than I'm, in type 1 and type 2 diabetes is not yet solved. Why? Because we have endpoint analysis. And having the stem cell system, you can understand etiology of disease. But now coming to the question, how can we use these cells for cell replacement therapy? And here, I mentioned it. I think we have now a source. We have, you know, the donor problem, so to say, the organ donor problem can be solved via the stem cell technology. But it's still, the costs are too high, and the immune rejection is the biggest problem, the biggest hurdle nowadays. In diabetes, you could think of beta cells, you can place them anywhere in the body. So you could think of, you encapsulate the cells and put them anywhere, then they are shielded. But that approach currently doesn't work because it's also a diffusion barrier. So the cells have to measure glucose. They need to be close to the bloodstream. And so that is a technical challenge. But I think if material scientists, immunologists, and stem cell biologists come together, we probably can overcome some of these hurdles.
Thanks.
We want to shut.
Yeah, it's already time up,
All right.
but it's, we could go on, I guess. But is anybody in the audience who would like to address a question? Because I think you were so well prepared. Thank you.
That's fine.
Is this
Thank you very
still
much.
a burning question you have, or?
No, it's fine. I'm fine. Thank you very much.
Thanks.
Thank you.
Thank you.
Okay. In 2019 in Germany, we celebrated the Bauhaus, which was an architecture and design school, very famous. And one of their famous wordings or sayings is form follow functions. And the idea was that form follow functions, but from the form you can learn about the function. So this definitely applies for proteins. And different shapes, you know, the way how they folded can mean it's either health or disease. So what we will talk about now is breaking the wall of protein analytics. And I do welcome Dr. Valentina Cappelletti. She's a research associate at the Institute of Molecular Systems Biology in Zurich in the Polar Picotti Lab. And there she investigates how the 3D structure of proteins controls their function in living systems. Her work extends the structure is function axiom of molecular biology to the systems level, enabling systems-wide structural proteomic analysis in health and disease. So 2 diseases she is thinking of with her research are Alzheimer's and Parkinson's, where incorrectly folded proteins play a major role. So she provides a new method, a proof of principle, where she can understand structural changes of proteins in an organism, which might lead to important breakthroughs to fight diseases. Her matching partner is Professor Markus Ralse. He's the director of the Institute of Biochemistry here at the Charité, and Markus is known for his discoveries regarding the coordination of dynamics within the cellular metabolic network and the development of analytical methods to tackle complex biological question. So Markus has been a group leader at the Francis Crick Institute in London since 2013, and 5 years later he became Einstein Professor of Biochemistry here in Berlin. So he will give us his thoughts on Valentina's concept and guide us through the discussion with her. So warm welcome for Valentina.
So, thanks, everyone. I'm honored today to be here to show you the method that we developed in Paola Picotti's lab to study protein structural alterations. And I will start from a very basic explanation of what a protein is. I know that this must be a bit boring for some of us, especially who's very specialist in protein structure. So I promise that the last part of the talk will be a bit more interesting for those of you. So proteins are the building block of life. They form our cell, tissues, and organs, and serve many important roles in our growth, development, and everyday functioning. For example, they work as antibodies, protecting us against invading pathogens, bacteria, or viruses, like coronavirus, for instance. They also work as a hormone, transmitting signals in our body. And this is the example of oxytocin, which regulates our social behavior. So the central importance of protein is also reflected by their name, that maybe you don't know, but is derived from the Greek word proteios, which means primary or in the lead. The whole set of proteins in an organism is called proteome, and it has been estimated that a human cell contains from 1 to 3 billions of proteins, so a huge number. And this makes challenging to study biological systems and how they work. To give you an idea of the complexity of a cellular biological system, this is how a cell inside looks like. So it's a crowded and dynamic environment full of different molecules, and each molecule has its own shape and size. And among those molecules, proteins are the most diverse one. So how does a protein look like? Here we are. This is a 3D representation of a human protein. So protein form complex and fascinating structure, like this one, for example. And they come with different size and shapes. And inside the cell, they can exist alone or in complex with other proteins. And they can also change their shape dynamically and frequently. This protein here is a bacterial protein. It's called HSP90 and helps bacteria to cope with heat. So I want here to make an example of how the shape of an object is important for its function. Think, for example, how the circular shape of wheels ensure that a car can move smoothly. Similarly, the structure of a protein, it's important for its function. And this is the so-called structure to function paradigm. So here an example. The structure of a protein is, for example, important for a protein to bind another molecule. So thanks to this structure, the molecule can bind the protein and activate it, exactly like a key opens a lock. But sometimes proteins fail to acquire the right shape, and this can be due to many reasons. Genetic mutation, changes in the intracellular environment. And when this happens, when there is this structural alteration, it's possible that the protein is not able to work anymore, like not being able to host its partner anymore. And misfolded or incorrectly folded protein have been linked to a number of human diseases, especially neurodegenerative diseases like Parkinson and Alzheimer. In the 1920s, Frederick Lewis first described the presence of protein aggregate inclusions in the brains of patients with Parkinson's disease. Inside those structures, later called Lewy bodies, researchers found a protein, alpha-synuclein, which in those conditions acquire an unusual or aggregated shape. So Lewy bodies are not present in healthy individuals, but the interesting thing is that alpha-synuclein is present in healthy individuals too. It's present in all of us, just with a different shape, not in this aggregated form. So again, this tells us how important is the structure of a protein for its function. Almost all biological processes are regulated by different molecular events, which involve a structural change, a protein structural alteration. For example, when a protein binds to a small molecule, there is a structural change. The same happens for protein chemical modification, like phosphorylation. Or when a protein binds to another protein, or to DNA or RNA, or upon aggregation. But how can we study all these structural events as they take place in a cell? Well, many biophysical methods have been developed over the years to study protein structural alteration in vitro, and we heard some of them. But they are not applicable to a whole proteome, meaning to the hundreds or thousands of proteins as they may be changing in a cell upon certain perturbation. So to overcome this limitation, we developed a new molecular technique called LIP-MS, which stands for Limited Proteolysis Coupled Mass Spectrometry. And this technique allows us to study protein structural alterations for many proteins at the same time, simultaneously, and within their physiological context. So from this structural information, we can also derive information about the function of this protein in this particular context. Let me put our research in the context of other recent scientific breakthroughs. So in June 2000, the Human Genome Project made it possible to read the whole collection of human genes. 14 years later, it became possible to read the whole collection of human proteins too. While this is a powerful approach, it has a major flaw. It is blind to the structure of proteins, but only tells us how much of each protein is present. And here is exactly where our technique comes in. We can now measure protein structural alteration for many proteins at the same time and compare, for example, healthy and disease condition and identifying proteins which undergo a certain structural alteration in those diseases and eventually get some information about the molecular mechanism at the basis of the disease. Now you may wonder how our method works. Here we are. Let's say that we have two conditions that we want to compare. So from one side, we have cell grown at normal temperature. On the other side, we have cell grown at high temperature. And let's say that this perturbation or this change in temperature cause some proteins to change their structure. And for example, they pass from an open or more relaxed conformation to a more structured one. So what we do, we digest our proteins with a molecular scissor. In this case, an unspecific protease. And this protease will cut the proteins based on their structure, meaning that the cut of the protease will reflect the accessibility of the protein to the protease. So, if a protein is in a more relaxed state or open, well, then the protease will be able to access it easy and will cut it in many fragments or peptides. But if this increase in temperature causes this protein to aggregate, then the protease will not access it that easy and will generate a different set of fragments. Those peptides can be measured by a technique called mass spectrometry, and later Marcus will tell us more about this, to generate fingerprints or barcodes reflecting the structure of the structural alteration of this protein in this particular condition. So not only we can identify which are the proteins that have a certain structural alteration after this perturbation, but we can also identify exactly where in the structure of the protein there is this alteration. And this can be used to generate mechanistic hypotheses about the function of this protein in this particular context. LipMS can be applied to any organism of interest, from small bacteria to human cell, to generate a vast amount of data. So we can apply it to measure usually thousands of proteins at the same time. But let me give you another example of how the method works. Let's say that we have a protein which binds to another molecule. So the red molecule is inside the protein in the active site. So with our technique, we are able to identify these the structural change that derived by which is the consequence of the binding of this small molecule in the active site. And we can localize exactly in the 3D space where the structural alteration takes place. And this is exactly the information that we need to infer functional information. So to make a mechanistic hypothesis about the functional state of the protein in this context. In this case, the protein is bound to a partner molecule, so it might be in an active state. Of course, all this structural information that we generate can be connected together to generate an integrated view of how a system works. But which are the possible applications of this method? How are we applying the method? So first of all, we applied LipMS to study protein structural alteration as consequence of different perturbation, genetic perturbation, environmental, like metabolic perturbation. But we also apply LipMS to study to identify drug targets or study drug mechanism of action. We are currently applying LipMS to study protein aggregation, especially in the context of aging. And recently, we applied LipMS to study and to find structural biomarkers for disease, in particular Parkinson's disease. So I'll quickly show you how this works. So we can collect blood samples directly from healthy and diseased individuals. We can apply LipMS directly on the blood samples to identify proteins which have certain structural alteration as consequence of this pathology. So this information can already be used to as a biomarker, so for the early and accurate diagnosis of the disease. But it's not only this. We can also look in those proteins where the structural alteration takes place. Well, if we have a structure of this protein, of course. And this information can be very helpful for us to make hypotheses about the molecular mechanism at the basis of the disease. So what's next? I've shown you that we applied LipMS to identify structural biomarkers for Parkinson's disease. But this approach can be applied to any condition or disease of interest, from cancer to antibiotic resistance. So we think that implications of our method are many. Of course, artificial intelligence is opening up big opportunities in biomedical research. So we are integrating deep learning technologies with our method to investigate the vast amount of data that we are generating in the optic of generating valuable knowledge in the field of personalized medicine. And here I just want to make an example. Let's say that we have patients not responding to a certain drug, or in case a drug shows some strong side effects. Well, we could use LipMS directly on samples from those individuals to identify drug mechanism of action. And this information can potentially be used for to design personalized treatments. So in summary, and to conclude, I show you how we moved from the human proteome to a new era, where we can now study the dynamic structurome, bridging, let's say, holistic and mechanistic understanding of biological systems. With this, I would like to thank you, and I'm very happy to discuss this with Marcus and with all of you, if you have questions.
What a service, no? I can get used to it. Thank you so much. Thank you very much, Valentina, for this very nice working down a technological talk for a very general audience. Could I have my slides, please? Fantastic. Great. So as we have seen today, very often scientific breakthroughs are very much bound to technologies which become available and to technological breakthroughs. And I couldn't resist to put in a quote here from Fred Sanger, who said that very often technologies are very important, let's say the least. No? And Fred Sanger must have known it because he got his first Nobel Prize for finding out to sequence proteins at a time where most people couldn't even agree that proteins have a sequence. And then later, the same individual figured out how to sequence DNA. And my previous supervisor, Steve Oliver, once asked Fred, hey, Fred, how did you come to the idea to sequence DNA? And his answer was, well, I knew it's an important problem. And then I went to the lab and thought about it every day for 7 years. Yeah? So sometimes it really takes a long time. Now, we have also seen this today. So several of the breakthroughs we heard today, they were attached to technological innovation. First talk used Cryo-M as a big technological innovation, which now allows to solve structures we couldn't solve before. We have heard about CRISPR, Cas9 this morning, which opens new avenues. In the previous talk, it's a technological innovation. Before we had single cell sequencing. So to all of those breakthroughs, we had technologies were involved. Now, sometimes these innovations are very noisy, and we all talk about it. Optogenetics, an example today. CRISPR-Cas was something of those. And those are very important because sometimes these trigger hypes. We in science are a little bit susceptible to hypes because of our short-term funding strategies. But there are also technological innovations which are a little bit more silent, but they're equally transformative. And that's about one I would like to talk today. I get a little bit of sunshine. That's also nice here. I hope you can still see the slides. And this is about mass spectrometry. As Valentina did allude to, mass spectrometry is a technology which comes into biosciences, biomedical science very strongly at the moment. But this is a long development. I would like to explain why is it so transformative. Yeah? So a lot in biomedical sciences deals around proteins, because we have heard proteins are the molecular functions, they function in a cell, but they never do this alone. Yeah? Proteins interact with all sorts of other biomolecules in order to function. They interact obviously with themselves, they do protein-protein interactions, they interact with nucleic acids, they interact with membranes, they interact with small molecules, they interact with metabolites, they interact with drugs. And all of those interactions, they influence how proteins function. And I have a simple example for you to illustrate this. Glycolysis is the metabolic pathway that we study the longest. Glycolysis research actually dates back to Justus van Liebig, who was fighting with Pasteur in France, and they banged their head of whether this is created by living organisms or not. And we have a pathway map now since 70 years. And still after 70 years, we know that 10 enzymes are implicated, and we have not a single mathematical representation that fully captures the behavior of glycolysis. And the reason is that we really struggle very often to understand with the way we do experiments how those proteins behave in a living cell. And this is a bit more complicated than it seems first. So Valentina had a nice picture of a crowded cell, much bigger than this one. So in the cell, there are a lot of those interacting molecules which surround an enzyme. But the environment that one cell has is also influenced by the surrounding cells to that cell. So that's the only picture here from my work. These are some microbial cells. They interact here. They exchange metabolites in order to live together on a media, which is here in black, where they couldn't live on their own. But the fact is, if they cooperate together metabolically, they change their own metabolism. They influence the intracellular environment from each cell. And therefore, in each of those cells, the same protein functions slightly different. Okay, so now that creates a problem because many of the technologies that we have available, at least on the structural space, on the molecular space, they required us to work with one metabolite, with one enzyme, with one structure alone. Okay, this doesn't mean that this is not good, because of course there was a tremendous progress with all those technologies we have available. We hear about many drugs that worked, we heard about many diseases that got cured, but at the same time, we also know that part of this success is always been possible because we could connect this molecular information with other information. Genetics is extremely important. I have another example for you. If we just have the structure and not have the genetics, how difficult it can be. I did my own PhD on a disease called spinocerebellar ataxia type 2, and this is part of a series of neurodegenerative disorders, and they're caused by mutations in those proteins, and we know the protein, we know the gene, and decades longer, still nobody can say in most cases what those proteins really do. Yeah? So we need to integrate a lot of information at the moment to find out. So better would be to understand how those proteins function in a cell and get to the molecular components. And this is now where mass spectrometry comes heavily in. So mass spectrometry is not a new technology at all. It also has been developed over many, many decades. It became a discipline probably some 50 years ago. There is a picture from mass spectrometrists from the early 1970s, from a picture that I took from the Chemical Heritage Foundation. And it has continuously been improved ever since. And what does it allow to do now? It now can be used to quantify not just one molecule or 5, but you can quantify tens or hundreds of thousands of molecules. If you nowadays record a cellular proteome on a mass spectrometer, probably you're talking about 120 to 140,000 tryptic peptides that are quantified in a single cell. Not only can you identify those peptides, you can also quantify those peptides. Not only can you quantify those peptides, you can also meanwhile search for modifications that sit on those peptides. You can meanwhile also measure turnover rates of those peptides, so means you can measure how fast they are produced, how fast they are consumed. There are new ideas how you can now localize those peptides in the cells. You know where they are made, how long they are made, how fast they are turned over, and where they are. And meanwhile, we can more and more also look where those peptides and the proteins they come from, how do they interact. So Valentina's talk was in that area, so it's very important that we can get to methods which allow us to find and trace those interactions. So what is beautiful about mass spectrometry, it's not discriminatory to which species you put on it. Yeah, you can measure a plant cell, a fungal cell, a bacterial cell, a mammalian cell without a big change in the technology. The costs are low. That's also very nice. Mass spectrometers cost a bit money. They're not as expensive as a beautiful cryo-EM, but each sample you run on them is relatively cheap. So we are talking about in high throughput mass spectrometry now on measurement costs of 5 to 10 euros. And you can make those methods also to move to the clinic. Yeah? And this is step by step now changing every single experimentation that we know in the biosciences and will very soon do for diagnostics. Great. With this, I come already to the end of my talk and hope there will be many questions to Valentina and to the field as a whole.
Thank you for that, Markus. It was very lively.
Valentina, let me start with a question to you. We all have been discussing a lot another breakthrough recently, which is the possibility to predict protein structures from the primary sequence, to use AlphaFold. So this is, of course, not yet at the level of quantifying intercellular dynamics, but clearly it's probably getting there. So what are your takes on this, and how useful is this for your?
Yeah, so AlphaFold, it's just game changer. So maybe for those of you who are not familiar, AlphaFold is a new program from DeepMind of Google, which allow us, so new algorithm, to predict protein structure from sequence. So you can imagine that in our technique, we need to have information about structure. We don't measure structure. This is done with other technique, with cryo-EM, for example. We just measure structural alteration. So we need a structure to know where this alteration takes place and to translate this into a functional information. So AlphaFold, in this sense, is helping a lot, because now for like almost all the proteins that we measure, we have a structure. Of course, it's a predicted structure. The quality is pretty high and close to experimental data. Still, we don't have information about many different conformations of the same protein, or many different, like, for example, we need information about how is the structure of a protein when it binds to another molecule. And this is still something that we need to, let's say, we need to integrate. AlphaFold can be used when we don't have other experimental data. So I think it will also come. So I expect this to be to be one of the next step of AlphaFold. Is already predicting multimer, so not only monomeric protein. So it will probably come, but still we need experimental data. But anyway, it's like a big step, and it was a game changer for our field and for our technique. So.
Christiane.
Great talk, Valentina. When you look at the proteins, do you see a difference between the structured part of the proteins, and many of these proteins have intrinsically disordered regions, which may be more dynamic. So do you really see this in your data, and what can you say us about
Yeah, it's
this?
a great point. So, yes, so we are... as I said, we use an unspecific protease. So the idea is that this protease will cut based on the structure of the protein and not the sequence. The proteinase K that we use has some preferences. So we see that indeed unstructured regions are more frequently probed by our technique. So what we would like to do now is to understand better the behavior of the protease that we use, proteinase K, for example, to be able somehow to, without, let's say, any previous knowledge about the presence of unstructured region, to be able, from the data that we get, to infer information about, yes, the presence, for example, of unstructured region. So for sure there is, so I think that we have a good readout also to at least have probability about the presence of unstructured region. But for sure, those are the regions that we tend to see the most in our data set. Not the only one. I think we can play or fine-tune the experimental condition to adjust it in a way that if we want to see more structured region, then we will have condition where we digest for like a long time. And this we know that will enrich our sample for those structured regions. But if we want to detect or probe unstructured region, well, then those are the conditions that we usually do. So we do a very short incubation with our protease. And so there we know that most preferentially, and this is what we see, we probe unstructured region, yes.
There was a question here.
Thank you. It's more of a remark. So the AlphaFold algorithm, the fold prediction, relies on the protein data bank, which gives us only the most stable of folds. But life is made possible by the inherent flexibility of biological systems, by molecular machines. So work like yours and integrative structural biology, in my opinion, is really the new frontier. We need to understand not only the rigid structure, but the structure of macromolecules in movement.
Yeah, that's a great point. So when we define our, let's say, breakthroughs as dynamic structurum, that's exactly with a word maybe not that easy, but we wanted to point out that indeed we try to add the dynamic information to exactly to this field. So we try to, with our readout, to reflect how a system can adjust quickly and dynamically. So what we have, it's a picture of a certain organism in a certain moment. It's not a static image. So it's something, it's challenging from one point of view, because of course it's something that takes, it's not that easy for the interpretation, but of course it's a very rich amount of data that we have, and of course it's taking into account the dynamicity of systems. Yeah. Thank you for...
Live Sander, I think, was next here.
First of all, I want to applaud you not only on your work, but also on the really beautiful and very excessive presentation. Thank you very much. I have a rather naive question as a layman. So the method that you used reminds me a little bit of DNA's protection that you do to study chromatin structure. So could you use that, for instance? And the next talk is about antibodies targeting certain regions of the proteins. Very often we don't know where these bind, right? So we have very potent antibodies that. So could you use that to sort of predict where an antibody would bind?
Yes, that's exactly what we do too. Yes, it's a great point. Yes, exactly. So what we can do is we have data on that already. It works very well. So we can identify exactly where an antibodies bind, and we can think of using this information to develop structural antibodies. So antibodies is not only specific per certain protein, but for a protein with a certain conformation. So yes, definitely we are also going in this direction. It's a great point.
Patrick?
Yeah, but then you need the antigen information, right? The antigen information you need. When you no?
You just need the antibody. You
okay, I have another question. What is with the resolution of this method, Is it possible to find very tiny differences, for example, on amino acid level, to recognize maybe a binding pattern for an individual drug, and maybe also the changes, the selectivity of individual drugs? What do you think about this?
Exactly. Yes. The resolution is few amino acids. So we can really identify exactly where a drug or a molecule binds. We are speaking about 6-7 amino acids. So we have a very specific region of the protein that will change because of the binding of something, and we can just capture that. Yes, so...
Okay, I think it's time for one more question. Daniel?
Yes, please.
Is there a reason why you focus on one protease? You said protease K. Would you get more information if you would, test a whole variety of proteases, which different digestion patterns?
Yeah, of course. So, and that was done at the beginning of the method when we develop it. So. Yes, it can be done. You can use additional one. This is the one that gave us the best result and was more also stable in the experimental condition that we use. But there is no limit to the fact that you can use any other protease. I just want to mention that we do a second cut after the unspecific protease, usually, which is done with trypsin, and this allows us to of course, help, to have a pattern that we can then use for the identification. It's a bit specific, sorry, for the identification of peptide. But you can also use an unspecific search. So you can also avoid this step and just use the protease of interest. We tried thermolyze and we tried other. And yes, it just depends on, I think, the question that you have and experimental condition that you want to have.
Great, I think in sake of time, we need to move on. Thank you, Valentina.
I think we'll see this method flying. So I think what we learned from the corona pandemic is that we cannot take anything for granted in life. So we cannot take for granted that we do have US guests here today from Kenya. So traveling is difficult at these times, and we cannot take for granted that people take their time to be here. I think it's great. So because what we're talking about now is new ways of treating COVID, and we're breaking the wall to treating COVID-19 through camelid nanobodies. So this is about alpacas and llamas. And so we have two people here today. So it's Dr. Florian Ingo Schmidt. He is one of the winners. He's a biochemist. And during his PhD at the ETH Zurich, he investigated the entry of viruses into host cells. So in 2013, Florian joined the Whitehead Institute at MIT to further develop the nanobody technology and apply it to better understand why virus infections and innate immune responses. Since 2017, he's leading his own research group at the University of Bonn, and together with Paul Albert Koenig, he set up a core facility for nanobody discovery, and llamas and alpacas play an important role. So Schmidt and Koenig developed nanobodies which attack the coronavirus and destroy the spike protein, and Florian will tell us more about this. So, and his matching partner from the Charité is Dr. Leif Erik Sander. He's a medical doctor. He's an expert in internal medicine and in lung diseases. And so he's now focusing on infectious diseases and vaccine research here at the Charité. So he's a clinician scientist. And with his team at Sander Lab, he aims to decipher host and pathogen interactions in humans and is particularly interested in the earliest events of the immune response and how they shape protective immunity. Currently, Leif and his team focus on dissecting human immune responses to SARS-CoV-2 and how they impact severity of COVID-19 and the generation of protective immunity. He will guide us through the last century a little bit and relate to giants on whose shoulders we stand when talking about vaccine research here at the Charité. So please, Florian, and later on Leif will take over.
Okay, good afternoon, everyone, from my side as well. And thank you, Christiane, for the kind introduction and for the organizers of the Falling Walls conference and the Charité for inviting me and giving me the chance to present some of our humble contribution to our fight against SARS-CoV-2. And I should, before I go into any details, point out that this is a shared research project not only led by Paul Albert Koenig and me, but also that involved more than 30 other scientists from all over the world, and including other species, as you'll see in a minute. And I should also emphasize that this is really a project that was building on quite serendipitous findings that were made and basically set the stage for the nanobody field. And as perhaps you have seen, there's quite a few efforts in the same direction that all demonstrate the same thing, that this field is really ready to be applied in the clinics. A virus is actually not much more than a vehicle and its main purpose is to really transfer a viral genome from one cell to the next. And in order to do that, it has to overcome quite some barriers. And viruses that are surrounded by a lipid bilayer have found a quite elegant way to overcome the most important barrier, which is the plasma membrane of the cell. And they have basically evolved proteins that can catalyze the fusion of the virus with either the plasma membrane or the limiting membrane of endosomes to really reach the site of replication. This very process can often be targeted by antibodies that our immune system generates to fight infections and prevent further spread. And usually neutralizing antibodies can either block the binding step or interfere with the fusion process itself. And in most cases, when we looked at these processes in more detail, we've also learned more about virus fusion itself, which is a quite complex process catalyzed by molecular machines. And we actually set out to do exactly the same thing for SARS-CoV-2. And as you know, SARS-CoV-2 has one major surface protein, the spike protein, which is a trimer and actually is able to catalyze the fusion of viral membranes with cellular membranes. And in a quite complex process, this trimeric protein here binds to the receptor ACE2, then undergoes a quite dramatic series of conformational changes which allow a hydrophobic patch of the receptor to insert into the opposing membrane and then undergo further changes that bring the viral membrane and the cellular membrane in close proximity and ultimately enforce fusion. In order to understand this process better, we took advantage of a curious type of antibodies that quite serendipitously during a student lab course was identified in Belgium in the 1990s, where students found that all camelid species, in addition to the conventional antibodies, contain also these so-called heavy chain-only antibodies, which are comprised of two identical heavy chains, of which a single variable domain is sufficient for antigen binding. And importantly, if you take this very single domain here that we then and express it as a single protein, which you typically call a nanobody, it usually retains the affinity for the target and can be used for many applications in research. And in the interest of time, I will just highlight a few here. So like antibodies, we here can generate nanobodies with the help of a functioning immune system that already basically selects the best molecules that bind with high specificity and affinity to their target. They, unlike antibodies, however, can be a lot easier produced in bacteria and yeast. They can be engineered to form multivalent proteins, and they can be identified in well-established techniques such as phage display. And even though their serum half-life, if used as a small molecule, is shorter than that of antibodies, this can also be engineered to match the properties of antibodies. So how did we raise nanobodies against SARS-CoV-2 spike? So we purified the receptor binding domain and then injected that protein 6 times into one alpaca and one llama, and then took a blood sample which contained the B cells that encoded for these heavy chain-only antibodies. And then we extracted the genetic information for all the nanobodies that were encoded in these blood samples and cloned it into a plasmid library, which we could then use screen using an established technique called phage display to identify nanobodies based on binding to their target. And this led to the identification of more than 30 different nanobodies, of which 8 showed the highest affinity when tested in the lab in so-called ELISA assays. In order to test if these nanobodies could interfere with SARS-CoV-2 infection, we made use of a system that allows us to do these experiments without going to the BSL-3 lab, without working with authentic SARS-CoV-2. And here we basically took a lab pet virus, vesicular stomatitis virus, and modified it in such ways that it basically no longer encodes its own surface protein and is dependent on the surface protein of SARS-CoV-2 to be infectious. And the virus also encodes GFP so that infected cells can be easily quantified by counting the GFP positive cells. And we did this and then pre-incubated the virus with the different nanobodies and found that 4 of our nanobodies were very potently able to inhibit virus infection. And this is typically measured as the IC50, the concentration at which half the infectivity is lost. And these results, in collaboration with Beate Kümmerer, could also be confirmed with authentic virus. And this largely supports what we've also known from the literature, that this pseudotype virus system very well recapitulates genuine SARS-CoV-2 infection. In collaboration with the team of Nicholas Wu at the Scripps Institute, we then solved the structure of all the neutralizing nanobodies in complex with the receptor binding domain of the SARS-CoV-2 spike protein and found that they bind to 2 different epitopes. One totally overlaps with the binding site for ACE2, which already explains how this is able to interfere with infection, and the other 3 bound to a site that was more tangential. Then, in collaboration with the lab of Martin Hellberg at the Karolinska Institute, we also solved the cryo-EM structure of the nanobodies here in complex with the trimeric entire spike protein. And what you can already realize on this image here is that the binding of nanobody E here to this trimeric spike brings the three different nanobodies in very close proximity. And this allowed us to do one trick that is much easier with nanobodies than with conventional antibodies, and this is to make multivalent nanobodies, in which we coupled 2 or 3 nanobodies in a row linked by a flexible peptide linker. And when we tested these nanobodies for neutralization, we found that the potency was up to 100-fold better, suggesting that these really make a much better tool to interfere with infection. However, as we all know, viruses are very quick at evolving, and particularly for chronic viruses, we already knew that viruses basically are able to overcome almost any drug by single point mutations. So in order to speed up evolution, because we had already learned earlier today that SARS-CoV-2 has a pretty good polymerase that at least minimizes the accumulation of mutations, we constructed an evolutionary system that allows us to study SARS-CoV-2 evolution in the test tube by, again, making use of this pet virus we use in the lab, vesicular stomatitis virus. And here we basically made a virus that was replication competent, and instead of its own fusion protein contained the SARS-CoV-2 fusion protein. So we again generate a virus that is dependent on the spike protein for replication, but in this case can basically generate progeny virus that can accumulate mutations in the spike protein. And when we enforce virus replication in the presence of these nanobodies, we found that in all the cases of the single nanobodies, but also the trimeric EEE nanobody, that almost all the virus spike proteins underwent mutations that allow them to escape neutralization. So in order to overcome that, we took a different strategy. Now we basically linked 2 different nanobodies that bind to different sites on the spike protein by a flexible linker to generate, again, multivalent nanobodies, which also were at least up to 50-fold better than the individual nanobodies themselves. And when we treated these replication-competent viruses with these bivalent constructs here, targeting 2 surfaces at the same time, we found that this double hit that the virus took didn't allow any emergence of escape variants in the time frame we looked at. And back at those days, we were worried about the emerging of escape variants in an infected patients. However, now we're smarter. We know the bigger problem is actually that throughout the human population, we accumulate mutations that in part have been selected to overcome human antibody responses. So we also tested our nanobodies against all the emerging variants, and we are pleased to report that because they bind to 2 different interfaces, one of them, which is actually very rarely targeted by human antibodies, all the emerging variants are still very well neutralized by our bivalent nanobodies. We could also confirm in vivo experiments that when these nanobodies were systemically given to mice, we were able to prevent lethal infection in a mouse model of SARS-CoV-2. And again, this worked out well for the wild-type virus, but also for emerging variants. I should point out here that because nanobodies are so small, they have a very short serum half-life, so we had to use a trick here to keep them in the system longer. And in this case, we fused the nanobodies to an albumin binding domain, which hooks up the nanobody to the serum albumin and therefore couples it to the half-life of this molecule, which is about 30 days. In order to make a difference in the clinical approach against SARS-CoV-2, where antibodies had already been improved, and a bottleneck was rather the complicated way of applying antibodies as an infusion and the high price of production. We took a different strategy and tested whether nanobodies could also be produced without these half-time extending modifications and be applied as an inhaled molecule directly into the lung. And I'm showing you data here where we basically subjected nanobodies to this so-called nebulization process to basically produce a fine nebula of small droplets containing the nanobodies, which can be very deeply inhaled into the lung. And we also found that this nebulization process, which often harms more complex proteins such as antibodies, did not really alter any of the structure of the nanobodies, didn't lead to aggregation, and also when tested functionally didn't compromise the neutralizing activity. And we envision that such application of nanobodies directly into the lung without any half-time extensions would be a very valuable addition to the treatment of infectious diseases. We also wondered about one thing when we looked at the structures of our SARS-CoV-2 nanobodies, and particularly the bivalent nanobodies that were so potent in neutralizing infection. And this is, we observed that the receptor binding domain that these nanobodies bind to were always in a conformation that we call the up conformation that is not typically found in the spike protein, but only when it is bound to its genuine receptor ACE2. So we wondered if binding of the nanobody to the spike protein has any effect on the spike protein beyond binding it and preventing it from binding to cells. And in order to test that, we generated recombinant cells that did not contain any ACE2 and stably expressed the spike protein at the surface. And just for easier visualizations, half of the cells were green and half of the cells were red. And then we added to these cells one of the most potent neutralizing nanobodies, VE, and then tested over time what happened. And there I think, need some of your help to start the movie. And what we did observe over time is that within hours, all the cells started to fuse with each other to form one gigantic Symposium, which is a macroscopic readout for the activation of the fusion machinery, which basically suggested that binding of the nanobody to the spike in the absence of its normal receptor induced the activation of the spike protein and basically caused the fusion of the individual cells with each other. And while it may seem counterintuitive that this is beneficial in a therapeutic setting, we concluded using additional experiments that what happens in an individual virus particle is that the bivalent nanobody here binds to the spike protein in its pre-fusion conformation and essentially tricks the protein into believing it already has found its receptor to trigger conformational changes that normally catalyze fusion but are fruitless in the absence of a target membrane. And this leaves an irreversibly changed and essentially destructed spike protein at the virus surface, which prevents the virion from infecting any further cells. To sum up, I think standing on the shoulders of giants, as pointed out earlier, our work contributed to the identification of a novel mechanism of virus neutralization, and we could nicely show that with actually quite limited protein evolution, we were able to develop tools that fulfill all the properties a nanobody or a protein would have to be used as an inhaled therapeutic to fight SARS-CoV-2 or future infectious diseases. Last but not least, I'd like to thank all the involved scientists, as I said, more than 30 scientists from all over the world, our funding sources, and you for your attention.
Thank you, and please, Live, take over.
Well, thank you very much, Florian. And of course, to the other speakers that have given us, insights into really the mechanics of how proteins interact and antibodies interact. And I do not work on antibodies or nanobodies, but was asked to sort of put Florian's work into perspective. And working at Charité, I could not resist of sort of taking you back in time when all of these nice technologies were not available. But I think people with quite limited technological means came up with extremely forward-thinking theories and talking about breakthroughs and standing on the shoulders of giants. I think we can honor some of those discoveries made actually here in Berlin more than 100 years ago.
So, and I think the overarching topic of Florian's work and the work of others that have tried to use antibodies or nanobodies is basically how can you transfer the state of immunity from few to many. So you have an individual that has acquired immunity to basically infection, and how can we utilize that capacity in order to immunize and make other people immune. So obviously we have the concept of active vaccination, and I'm just gonna briefly mention that. But in fact, what Florian's developing is tools to perform passive immunization, so to transfer immunity from one person to the other, or ideally to many others. And this is a drawing here that was made by Paul Ehrlich more than 100 years ago here at Charité, and it basically depicts very basic concepts of immunology long before any of these people were able to see or to measure all of these things that we heard about earlier. So the concept of transferring immunity, I think, really dates back to Emil von Behring and Dr. Kitasato. Because what they had noted was that there had been other attempts, but what they had noted is that animals such as rabbits that have been exposed to some of the really dangerous diseases at the time, diphtheria and tetanus. You know, diphtheria was a disease that killed 25,000 kids a year. You know, that was at the time, so a very, very serious disease. But animals that had survived exposure to this or survived exposure to the toxin. So this was a crazy prolific time in science, you have to say. At the same time in Paris, Pasteur, they had discovered that the toxins released by these bacteria were the ones that caused the disease. So you could also take the toxin, expose rabbits or other animals, and if they survived, those animals became immune. And Behring had speculated that it's a component in the cell-free fluid in the blood that is conferring the immunity, and that you could transfer that. And I think this is a landmark piece of work that was published in the Deutsche Medizinische Wochenschrift über das Zustandekommen der Diphtherie-Immunität und der Tetanus-Immunität in Tieren. So how immunity is brought about to tetanus and diphtheria. And what they basically state, and actually for the sake of this symposium, I went back and read all those old papers, and I can just recommend you in doing the same. It's really amazing. And so the immunity of rabbits and mice who have been immunized against tetanus is based on the capacity of the cell-free blood to inactivate the toxic substances. So you can take it and inactivate the toxin, and then the toxin, you can transfer that toxin, give it to another animal, and this other animal will no longer get sick. So it's inactivating the toxin. Of course, they didn't know how this is brought about, so they speculated they are antitoxins. And I think this is basically summing up their work. And you know, at the time there were not these nice figures and so on, that you had some tables and some statements, and you know, you'd have to believe the guys. And they were, you know, talking about other results other experimentalists had performed. But so in fact, these are the 4 claims that they make. The blood of tetanus immune rabbits possesses destroying capacities. It can destroy toxin, and this capacity is also detected in the serum, so in the cell-free fluid. And then came the crucial experiments. So this capacity is so durable in nature. So if you transfer the serum from one animal to the other animal will be protected. And this is basically the birth of serum therapy, and I'm going to show you in a second, which went on to be one of the major breakthroughs in fighting this deadly disease called diphtheria. And in fact, this inactivating capacity is lacking in animals that have not been exposed. So it's something that can be learned and that can be acquired. But the real progress actually in using this knowledge for therapy really came when Emil Behring at the time was collaborating with Paul Ehrlich, because Paul Ehrlich came up with a way of how to measure precisely the antitoxin activity in the serum and how to enrich that activity, because just transferring a little bit of serum wasn't enough. You had to enrich that activity. And through a series of experiments and again, landmark papers that the two of them published together, in the end came the Behringschen Heilserum, so a serum that was given to sick individuals suffering from diphtheria. And to ramp up production, he teamed up with a big company, Höchst, and then later on gave birth to his own company. And many people think that he actually cheated Paul Ehrlich out of a lot of the revenue of this joint venture. So what happened was, at the time, this is Emil von Behring here later on in his factories in Marburg. So they had stables with thousands of horses, and this was even going on into World War II, and where they immunized animals and took the serum of those animals, enriched that, and could give it to soldiers in the field against tetanus or to children and treat diphtheria. So, and Emil von Behring was actually awarded the first ever Nobel Prize in medicine for this discovery. Again, I think Paul Ehrlich was a bit disappointed, but then again, these people were so bright that they didn't settle on one major breakthrough in their lives. And Paul Ehrlich, I think, made 3 seminal discoveries, and for one of them, he was lucky enough to also get a Nobel Prize. So, and this is, I think, the one that is most beautiful if I think about immunology. So this is a theory that that Paul Ehrlich was hatching out in his office here. So imagine all your files on your computer stacked into one room. It's probably what it would look like, and so without the technologies that we heard about. He came up with a theory that actually was very much on point. So, because he speculated about a so-called side chain theory. So he said, when the organism faces toxins, these would be nutrients, and there are nutrients, and there are so-called side chains. So I guess he was talking about receptors that are on the surface of a cell, and then the nutrient combined, and the cell can utilize the nutrient. But then there are toxins, but there are also side chains on the surface of the cell that can specifically bind the toxins. And if there are more toxins, more side chains will be produced, and then the side chains will be produced in such an excess that the side chains are being released into the serum, and then that's what he refers to as antibodies. So he came up with this. And the only thing I guess that he got wrong, that nowadays we depict this interaction in the reverse order, right? The antigen would be the smaller one, and the antibody would be this bigger molecule here. But because these toxins look a little bit like antibodies, but basically these side chains here released from the surface of a cell. And if you think about it, this is probably what happens, right? B cells express B cell receptors on their surface. They get triggered, they internalize, and they release tons of antibodies. So this came about in the end of the 19th century, and this is I think one of the most beautiful and amazing theories that basically principles of it hold true to this date. So he went on, he was speculating. His next concept was the magic bullet, die Zauberkugel. Yeah. So he wanted to, and at the time he was thinking, how can we use those side chains to specifically target pathogens? And I think maybe also a little bit out of disappointment with Behring and so on, he went on to do other things, and he, on the side developed the first treatment for syphilis, and called that his first magic bullet. But in the end, people have gone back to this concept and said, the return of the magic bullet with the onset of monoclonal antibody therapies and so on. So. And what he said is that we have to learn how to aim chemically, so how to precisely fit to make a drug that exactly fits the pathogen. So he ended up also receiving the Nobel Prize, for instance, for this beautiful therapy. And next to him is Ilya Mechnikov, who happened to also be around at the time, and he discovered the first immune cell, which was the macrophage. And so the concept of phagocytosis was recognized at the time. So we have cells that can eat bacteria. But what the two of them didn't know is that the side chains, so the antibodies produced by B cells, also not known, they were discovered in the 1960s. B cells produce antibodies, and those are then bound by the macrophages on the surface and help them eat the bacteria. And they received the Nobel Prize together, not knowing that those two discoveries were so closely interlinked in 1908. So this is, and Florian showed you that this is how the side chains actually look like. This is an immunoglobulin, and you have this variable region. I'm not going to go into detail because Florian showed you all of the detail, but this is basically aiming chemically. This is the magic bullet concept of Paul Ehrlich, how you can target an antigen. So, but it took quite a while for this concept that Paul Ehrlich developed to really mature into what we think is happening today. And it was actually Macfarlane Burnet, so an immunologist at the Walter Eliza Hall Institute in Australia, who came up with a clonal selection theory, because there were some troubles with the concept of Behring, because the variety of molecules that it can use to generate side chains or antibody is too vast for it to be already there, ready to go in your repertoire, if you wish, of cells. And so Frank Macfarlane Burnet came out with a clonal selection theory in which individual cells that happen to be specific to a certain molecule can then clonally divide, and they can also change their genetic information in a way to better fit the target. And this is basically the foundation of the clonal selection theory, which obviously also at the time was a major breakthrough and was also awarded with the Nobel Prize. So you can see this whole history of antibodies is littered with Nobel Prizes and major breakthroughs. Why am I mentioning the clonal selection theory? Because nowadays what we're actually using a lot already in the clinic is monoclonal antibodies. So we're taking individual clones that produce specific side chains that chemically target the pathogen, and we can clone out an individual cell. And what has been used for decades is making hybridomas. So you take out individual plasma cells, and interestingly, plasma cells were also discovered in the 20s, and then you can fuse them with a tumor cell, and then you have immortalized cells that produce one specific antibody that is specific to your target, and you can use that for treatment. And I think what Florian is describing is basically technological advances to this quite ancient concept here. And we've also come a long way. And what I think many people are using nowadays is single-cell B-cell cloning. So you don't have to immunize a mouse, but you can take a person. We also did that in one paper at Charité. We took people that were convalescent from COVID and cloned out individual B-cells, and then you can express the heavy and the light chains, and you can make monoclonal antibodies that way. And I actually took this headline from a paper that came out, or a review that came out, so Return of the Magic Bullet. So you can see the first monoclonal antibody was made in 1975, and slowly from the 1990s, monoclonal antibodies have entered the clinics, and nowadays, if you're a clinician, what have you mAb is given in any given disease, right? There's so many different mAbs, but some of them are very, very well known, such as rituximab that depletes B cells, is used in treating B cell lymphomas, and many other antibodies. And again, antibodies targeting a specific surface, again, the magic bullet theory on the surface of tumor cells, have now entered the clinic as immunotherapy and were awarded the Nobel Prize, I think, 2 years ago for the checkpoint blockade, right? And I think what happened this year is that people went and cloned individual antibodies and selected the ones that bind the strongest to the SARS-CoV-2 virus, and they work beautifully. The thing is only that nobody knows about it, and this is why they're not used. But if you give these antibodies early on when you're sick, you can basically rescue 85% of patients. 85% of patients that receive monoclonal antibody early after SARS-CoV-2 infection do not have to go to the hospital and don't die. And the most famous patient who received this was the former President Trump, who received this early on, and he had a lot of risk factors, if you ask me. So, and this is the last slide. So what's the next step here? And I think that's also where nanobodies can come in very handsome, is that we can use those antibodies, again, with the Ehrlich's paradigm to target the pathogen, but we can link that, we can hook that up to other drugs that can help us fight the disease. And I think we've come a long way now with nanobodies on the way, and I think we've gone a little bit over time. Hope it was interesting, and thank you very much for your attention.
Thank you so much. I think, yes, we're a little bit over time, but I think it was an excellent journey through the last century. So, and I don't take it for granted that you actually dug deep into it. So many thanks. So we should finish now, but I think we should have a discussion. 5 minutes, right? Yeah? So, okay. How do we do this? So, Markus.
Live audience.
True, online. So could you use these nanobodies to inject something else into the cells to boost effectiveness, like a protein which triggers apoptosis to the receiving cell?
In principle, yes, although I have to say the most straightforward application of nanobodies is still extracellular targets. There's a lot of development to try and make nanobodies that find their way into the cell, and certainly you can make them be internalized and target the endolysosomal system, but to reach the cytosol, I think there's no broadly applicable method yet, although there's a lot of interesting research even going on in Berlin in that direction.
The SARS virus, the virus is internalized. Would it not take the antibody with it, and you could bring a second booster to it in the cell?
So you mean you could enhance uptake of variants? So that you could, in principle, and that was more a concept people were a little afraid of, that this may enhance infection as a process called antibody-dependent enhancement. In our particular case, we know that the nanobody, the way it modifies the target, also leads to the shedding of this bit that is bound, so it cannot happen, and we also don't observe any enhanced infection. But in other cases, that can be, for the good or the bad of the patient, occur.
So Patrick Scherer, I guess?
Yeah, maybe a technical question. I think that there are also other ways to develop nanobodies, for example, in yeast, with yeast libraries. It's developed by Andrew Kruse, for example, in Harvard University. Do you think we need maybe in the future no llamas and alpacas, or is there any advantage to this?
I would say at the moment, the main advantage is that we can't beat the immune system at making good antibodies. I think there is a lot of hope that synthetic libraries made these things easier. We can't also inject everything into an animal. We can't inject anything that would make the animal sick or even be a toxin, right? So there's a lot of interest in that, and what you have to overcome is that you just need to start with much, much larger numbers of molecules that you screen for. And that's now going in the right direction. And we'll see, I think it will always be complementary.
And I think Christian Spahn, he's over here. It's fine? Okay, then Daniel, last question.
Maybe an interesting question would be, why do you think llamas develop this special kind of antibodies? And, maybe also thinking about MERS and camels, is there, speculate a little bit on what do you think is the biological sense of those
I would love to give you an answer with scientific foundation, but we don't know. You can only speculate that it was evolved from the natural antibody gene, and it basically started some 15 million years ago in the last common ancestor of all camelids, and it was kept. So it basically at least is some advantage, or at least no strong disadvantage. But we don't. You can only speculate that there are pathogens that can be better fought by this. We know that because of the different geometry, they bind different kinds of epitopes. So nanobodies are better at recognizing folded proteins and distinguishing conformations and antibodies are maybe better at recognizing a peptide. So that could be part of it.
Well, if that's all, then...
Okay, so thanks everybody. We're having a break now. And I think we meet again at, let's say, 4:20 because we were a little late. And Live already mentioned something we will continue with because we will talk about checkpoint inhibitors, but from a different perspective, and it involves cosmology, astronomy, so that's going to be something I'm looking forward. And we're talking about malaria as well. So we have our guest from Kenya and the US after the coffee break. Enjoy it, and thanks again. So let's start the late session, although it's heartbreaking to interrupt you with your talks because we were hoping you would mix up in teams and talk about your research and get to know each other. So this is the final set, and as I said, it's with our guest from Kenya and the US. And so I've got a little bit of history as well for you. We're talking about breaking the wall to symbiont-based transmission, blocking malaria. And a Canadian called Timothy Weingart, he wrote a book a couple of years ago, The Mosquito, A Human History of Our Deadliest Predator. And so in his book, he writes down, I think he's from historic research, he wrote down how malaria changed the world. And so the question is, it's been around for so long and we still have no cure or any means to fight this deadly disease. And so what we're talking about now is new ways of fighting those mosquitoes by not killing them. So we have Dr. Jeremy Herren here. He was born in Switzerland, I understood, but he's flew in from Kenya. So he's one of the 10 winners today. He's an entomologist, so dealing with insects, and microbiologist. He's interested in the interactions between insect endosymbionts and their hosts, like bacteria and mosquitoes, which transmit malaria. And Jeremy's current goals are to develop endosymbiont-based strategies to control malaria in Africa. To meet this goal, Jeremy Herren has established a research facility at Nairobi, and recently he published an important paper in Nature Communications and will tell us more about his findings. So this is our winner, and he'll be joined by Professor Frank Mockenhaupt. He is acting director of the Institute of Tropical Medicine and International Health here at the Charité. And in his research, he focused a lot on malaria, e.g. drug resistance, one of the major problems with the infectious disease, or the role of human genetics in relation to malaria. So Frank will give us an oversight how important malaria control measures are to fight it. And we look at what has been done so far and what are we missing. And I can tell you, Frank's a little bit skeptic about Jeremy's work, but we come to a good way to see how we go with this. So welcome, Jeremy. Enjoy his talk.
Okay, thank you very much, Christiane, for the introduction, and it's just such an honor to be here amongst really such excellent scientists with really elaborate work. I've really enjoyed it over the course of the day, so I feel very privileged to be with you today. So yeah, I think I'll talk to you a bit about malaria and our research looking at new methods to block malaria transmission. I don't know if this is ready for my slides. Okay. So yeah, that's just the name of the project, but I'll get straight into kind of an introduction, which is very sort of big picture, and this leads on very well from what Christiane said. Malaria is really, really bad, and it's bad at the moment. You know, we still have huge economic costs. We still have, you know, about 400,000 people dying every year. But if you look at it even in the historical context, things get, you know, pretty shocking. And this slide just kind of shows the cumulative numbers of deaths caused by a number of different infectious agents and diseases. And you see malaria is really right up there at the top. Now, one of the reasons for that is it's been around for so very long. It's a disease which was infecting our ancestors, even before we became anatomically modern humans. But it is still something that is, here to this day, and we have obviously developed a number of ways to control it, but we've not managed to break this cycle. So yes, that's kind of the sort of shock factor. But I think we should also be a bit more enthusiastic about malaria and about the prospect of finally ridding ourselves of something. And this, as I say on the top of the slide, the worst enemy of all. I mean, this is, been with us, been killing people. It's been synonymous with death for so many of our ancestors that we should be potentially enthusiastic that this generation could be the generation that gets rid of this once and for all. And I think it's hard to say that wouldn't be like one of the greatest achievement of mankind, and it's up there with all the other things like even things that actually are probably a lot less important, like going into space and things like that. And we should be more enthusiastic about this. That will bring more funding to it, because it is really an issue that is potentially a little bit neglected, and people got excited about trying to get rid of malaria in the 60s and 70s, and it didn't happen. And I think the community feels maybe is still sort of reeling from that. But anyways, so going forward, I think we should really try and tackle this, and it'll be amazing if we can. So just sort of a very cartoonish overview of the malaria transmission cycle, which is really important for my talk. Obviously, you have mosquitoes as a really integral part. Mosquitoes infected with the Plasmodium parasite bite humans. The parasite then develops within those humans. They then get bitten by these mosquitoes, which fly off, infect another human, and so the cycle continues. And of course, within each of these hosts, the human host and the mosquito host, you have a number of different forms of the malaria parasite. So we have come up with a lot of interesting tools which have been very helpful in this fight. As many people know, actually the number of people dying from malaria has declined very drastically in recent years. The malaria development of malaria drugs is something which has been a really major achievement. So that the issue there though is that most malaria drugs are not breaking this cycle. So that's really what we need to do. As great as that has been, these have prevented people who have the parasite from dying or from getting severely sick, but have a relatively limited effect on this cycle, because people that are treated with most malaria drugs can actually still transmit the parasite. There have been other advances. So of course, bed nets, really the big one that has been responsible for huge gains in malaria. Very effective at blocking the transmission cycle, which is great. Unfortunately, people just can't live under nets the whole time. You still have a lot of residual malaria transmission. People are out doing what people need to do. There's also been a lot of energy devoted to killing the mosquito, and that has had pretty good effects. So if you get rid of the mosquito, you also do block the cycle, either by insecticides or removing habitats. This has a number of challenges. Mosquitoes are very resistant. They evolve resistance to a lot of the insecticides that we use to kill them. There's often a lot of off-target effects of these insecticides. They hurt the environment. So this is kind of where we are now. But what we really need to do, we need to look at interventions that are going to stop this cycle, that are going to break the cycle. So there's a lot of interesting things coming along. We have transmission-blocking drugs and vaccines. So these are strategies that would block infected individuals from being able to transmit their infection to mosquitoes. And the last sort of one that's very much what we focus on, this is strategies that target the parasite while it's in the mosquito. And so this is an area which I think for a long time was kind of neglected as a potential place to target the malaria parasite. And I hope that over the course of this talk, I can convince you that it's somewhere that we should devote some energy to. Okay, so a bit of very broad background here, but this is just to put our research, to give you some context. What's really important to remember, and I'm sure that or everyone is aware of this, but microbe infection doesn't always equal in disease. So there's a lot of microbes that do not cause disease. And often you get sort of a spectrum like this, let's say. So you have things that are highly pathogenic. They make their host very sick. Their host doesn't survive very long, and that's not necessarily beneficial for the microbe. So you think about things like Ebola, for example, right? They kill their host very quickly. On a large scale, they're not particularly successful. Actually, a lot of microbes are in this area, either the yellow or the green. They're not causing that much damage to their host. And these are the ones that we're very interested in. I think people will sort of know the gut microbiome is quite a good example of something that's quite benign. But there's a lot of other ones which we don't see them. They're really not detected. They're these sort of invisible pandemics, things that will spread. They're incredibly effective at spreading through a population of animals. You don't actually see them because you don't see a disease, and that's usually what we're looking for. So these are common in all animals, especially possibly common in insects. And it may be sort of special to insects. You get these sort of different distribution patterns, which, you have something that's pathogenic. Usually it's kind of everywhere in the insect. Probably the same in other animals as well. These sort of may be more in between ones. Often you kind of find them in the gut. And these ones which are sort of real symbionts and maybe even beneficial, this is maybe where the insects are a little bit different, or actually I think insects are more normal, but we are quite different in that we generally don't have microbes inside our bodies and inside our cells when we're healthy. But that's not the case for insects. A lot of very healthy insects have microbes spread out through their body, usually quite localized to certain tissues. And another interesting thing about these microbes is they can be transmitted from a mother to offspring. And so they really are able to stay within these insect populations, which often have a very short generation time. Okay, so this is an example of one of these. It's probably one of the greatest pandemics of life, really, is this microbe called Wolbachia. It is found in about 60% of all insects, and as many of you know, insects are kind of, most of what's alive. So this is a really successful bacteria. It is symbiotic. It's transmitted from mother mosquitoes to their offspring. So a great example of one of these kind of invisible pandemics. This also serves as a second example for me, is kind of a proof of concept, because actually this Wolbachia bacteria, which is found in so many insects, has also been used in a way to block transmission. So it makes those insects resistant to viruses. And so there's a number of new projects that have released or transferred this Wolbachia into major vectors of diseases and then released them. And that's actually having a major effect on dengue transmission. So this graph just shows pre-release and post-release. And this Wolbachia bacterium is having a major effect. So yeah, a dual example there of an invisible pandemic that actually can do some good. So now shifting to my research, and the key thing, I guess, here in terms of breaking walls is trying to break the wall between lab research of mosquitoes in sterile environments and really kind of understanding what's actually happening in the field. And so what we've done is I do most of my work in Western Kenya in an area which is very good for mosquitoes. There's a lot of malaria, and we can collect mosquitoes from their natural habitats and rear them in this type of environment, which is completely open to the wild. It's quite close to a sort of natural habitat. The water comes from the environment, and we can really have a much better picture of what these mosquitoes from the field are actually like. But of course, we're studying things which are, inside the cells of mosquitoes. They're inherently difficult to study. They're hidden from us. So that's where we needed to combine this with a sort of a more high-tech lab research infrastructure where we can do molecular work and we can see things which are not always easy to see. So that's basically where this all started, and this was our major discovery, was that these mosquitoes that come directly from the wild, from different habitats, they're all actually very different, even if they are all the same species. And one of the characteristics that is really different is that their ability to transmit malaria is actually very different. So we were able to feed them on blood that has malaria parasite, and find that there are these subpopulations which are not actually able to take up the parasite. So in this case, a subpopulation was completely unable to transmit the malaria, unable to take up the malaria parasite. It doesn't develop in these mosquitoes. And this is something that we showed of repeated experiments on looking at transmission. And we were able to figure out why that was. In this particular case, this was because they harbored this symbiont called Microsporidia MB, which is so an endosymbiont. It lives inside the mosquito cells. It's a distant relative of fungi. Microsporidia are a very, very weird group of eukaryotic parasites. And so we then, did this experiment over and over again, and were able to, show that this was, really what caused refractoriness to malaria. So that's basically the key discovery that we've been building on. We just wanted to know a lot more about this Microsporidia MB and see whether it had any potential to help us with this blocking of the malaria cycle. So as I mentioned previously, with a lot of symbionts, Microsporidia is actually vertically transmitted. So mother mosquitoes infect their offspring. This is a mosquito ovary, and this is sort of the egg of a mosquito that's developing. You see this mass of Microsporidia MB that's getting into the egg at that very, very early stage. So that's a good sign. It's also sexually transmitted. So this is it's actually the testes of a male mosquito, and these are, I guess, the male accessory glands, so something kind of like a prostate, and this is the ejaculatory duct, and you can see all these little dots in here are the Microsporidia, which gets into the mosquito's seminal secretions and is then transmitted to females when they mate. It didn't have any negative fitness effects. So this is something which has been in mosquitoes, Anopheles gambiae, one of the better studied insects. I mean, still there's a lot we don't know about it, but it wasn't discovered up until 2 years ago, and the main reason is it doesn't cause a disease. These mosquitoes are not at all sick, and they develop actually very well into adult mosquitoes, and it's then transmitted. So no negative fitness effects. We found that Microsporidia MB is across sub-Saharan Africa, so many different sites. The levels bounce up and down for reasons that we don't fully understand, but they do at some points reach relatively high levels of about 20 to 30% of mosquitoes. So what can we do with this? Well, the idea is that we have these natural fluctuations of something that blocks transmission. They are not quite high enough. We think maybe in this red zone is where we have a significant impact on this cycle. And so we need to find a way to sort of up the amplitude of these oscillations, potentially to release more microsporidia MB, or to change the environment in some way that microsporidia MB can spread better. And this is basically the big challenge for us now, is to figure out a viable strategy to get more microsporidia MB out there. That's kind of what we're working on. We've got these cages that look a bit like the wild, and we can release mosquitoes that are infected. We've now isolated spores, and we're hoping that we might be able to release these microsporidia MB spores and that they would spread. The challenge is really in these natural environments. The system works very well in cages, but obviously mosquitoes don't live in cages in the wild. So, yeah, this is really the next step for us, a viable dissemination strategy that would be sustainable and cost-effective. So, I think that's basically all. I think, obviously, I hope I've made the point that the cycle is really the key thing when we're talking about trying to get rid of this disease. Most microbes don't cause disease. We need to think more about them. Insect pandemics can affect the ability of mosquitoes to transmit disease. So, and hopefully, I've convinced you that it's worth investigating these for some kind of transformative impact. So thank you very much. Just to thank the team, in particular 2 students that really were instrumental, and now we've got a much bigger group, and hopefully we can really get to the bottom of some of these key questions. Thank you.
Thank you, Jeremy.
Thank you. Thank you for having me, and thanks for the great talk, Jeremy, and for the promise inherent in that. I would like to put this in the context of overall malaria control, and as you will see, overall malaria control is trivial, rude, and rough. And we definitely and urgently will need novel approaches to cope with the issues. Right. Okay, just the last 20 years of malaria have seen a lot of change. We, the world, managed to reduce morbidity and mortality. Mortality more than morbidity, but the number of cases and deaths still is very high. If you plot it to sub-Saharan Africa, here the prevalence, you see that in the last 20 years, the overall prevalence has declined in African children, which is the main group for malaria. But the absolute numbers have not, and that is due to 60% population growth in that period. So it's a dilution to some extent. But what you can see is that mortality has greatly been reduced. There are estimates that some 7 million deaths have been averted in the last 15 years. Potentially, this is largely due to the implementation of working antimalarials, implemented around 2005, the so-called artemisinin-based combination treatments, or ACTs. This was made possible by the availability of funds. Around 2000, interest in malaria restarted, and you see this is funds for malaria control, not research, control, that from several dozen millions up to 2010, the world global expenses reached 2.5 to 3 billion euros, or US dollars. And then, thanks to the financial crisis and other impacts, since then it has been plateauing at around, let's say, 3 billion US dollars. This is insufficient. This is half of what RBM, UNICEF, WHO estimates of being necessary to deliver the interventions that we do have, namely those. These are the 5 evidence-based interventions to control malaria. And you see it's not that rocket science, it's early diagnosis and treatment first. Early diagnosis nowadays for sub-Saharan African children is available for roughly 40%. That's a major achievement in the last 20 years. Like 10 years ago, it was 8%. Adequate and prompt treatment, yes, we do have the ACTs, and they are available for 80% plus. The single best intervention that we still have today is the insecticide-treated nets. They prevent malaria by, let's make it 70%, but they reduce all-cause pediatric mortality by 25%. There's no vaccine in malaria, no other intervention that is as good as this. Another beautiful tool is indoor residual spraying. Jerry mentioned that, using DDT or puritrides every 6 months. That is logistically challenging and financially challenging, so you don't do this on a Saturday afternoon. But if you do it, you basically can interrupt transmission. We see that in Rwanda, where we work, there's no malaria anymore because of IRS. Coverage is poor, as is coverage for the ITNs. And then there's a particular mixture of chemoprophylaxis and treatment for pregnant women that also has a low coverage of 1/3. There's more, of course. I just mentioned the two. There is an approach called seasonal malaria chemoprevention. For example, in the Sahel, there's transmission only for 2 or 3 months. So you don't need to cover the kids or the population for the whole year, but only for the transmission season by monthly treatment, for example, blind monthly treatment. And depending on the drug used, this can almost eliminate malaria during the transmission season. There is a vaccine which is imperfect, based on sporozoite antigen, and that has a protective efficacy of roughly 30 to 50% and is not yet implemented. And then there's a lot of other approaches like simple drainage or fish eating the larvae or transgenic Anopheles and others. Malaria control has a number of problems. First, that's a personal selection, is the health system itself and the limited capacity to deliver these interventions to the population in need. The intervention gap. This is, of course, related to the funding gap. And also, we need to talk about sustainability and stability. All these interventions need to be integrated horizontally into the health system. Otherwise, they will not be sustainable. And what we see now at the moment in the COVID pandemic is that a lot of malaria programs are being disrupted. And we anticipate to see increasing numbers of malaria cases and death in the next years. Stability is obvious. If there's an armed conflict, nobody will take care of malaria control. And then there are two biological threats. The one is insecticide resistance, which is spreading, and drug resistance. And I want to use drug resistance to link what Jeremy just said with a newly emerging threat. That is artemisinin resistance. So this map shows you the distribution of the molecular marker of artemisinin resistance, and you see that Southeast Asia is the hotspot. Markers. In Cambodia, these markers had been detected some 20 years ago. In the decade following, there was a low level of clinical resistance, but nowadays we have a high proportion of actual clinical treatment failure. Very unfortunately, this now seems to be beginning in East Africa, autochthonously. So the mutant lines which have been detected in Rwanda and Uganda are of local origin. We see the increase over time, and we and others in Uganda, Rwanda see that there are two major lines, parasite clones now. They are in vitro resistance. They show some mild degree of resistance already in patients, and they don't have fitness costs. So in a condition of drug pressure, and there is drug pressure, these parasites will spread, first locally, regionally, hopefully not beyond that. If artemisinin resistance spreads throughout sub-Saharan Africa, we will face a malaria disaster, maybe comparable to the excess mortality that we have experienced with chloroquine resistance in the 80s. So we need to combat that. And this is where Jeremy can help. Spreading resistance is spreading resistant parasites. And in the original epicenter of resistance in the greater Mekong region, the aim is to eliminate the resistant parasite by eliminating malaria. And they're halfway through. Malaria in sub-Saharan Africa is a bit more complex. I'm not too optimistic that this approach will work. So we could use the microsporidia to, let's call it, sterilize the vector, or simply enforce traditional vector control, or break resistance by adding a third partner drug to the artemisinin-based combination treatment. Or, going back 130 years, I'm also member of the Paul-Ehrlich fan club. 2 miles, is it upstream or downstream? I wouldn't know. This river, there still is, Moabit Hospital, which was the place where the first synthetic antimalarial, methylene blue, ever was used on patients. It worked okay. But there has been renewed interest, led by Heidelberg, showing that if you add methylene blue to an ACT, you don't do this to enhance efficacy. Not necessarily. You will find that the post-treatment presence of gametocytes in blood is greatly reduced, both on the microscopy and on the molecular level. Post-treatment gametocytes would be those parasite forms which transmit resistance. Yeah? This works, is similar effective as is primaquine, another drug, but the side effect pattern is unpleasant. But we need more drugs like this. So in total, yes, I'm skeptic, but I'm born optimistic. From my point of view, the major problem in malaria control is money and coverage, which is somehow interrelated. If we would bring all the interventions to 100% coverage, we would be 10 years ahead. But we need novel approaches like a working vaccine or the symbiont way of sterilizing the mosquito. So there is reason to be optimistic. However, this optimism is not that fresh anymore. Thank you.
Thank you, Frank. I think it's wonderful that because science is going back and forth, so both of you came up, like Leif and you, with Paul-Ehrlich. So obviously we cannot call him anymore, but we can look into his papers he's written many years ago. So are there any questions from the audience? Would you like to discuss with Jeremy a little bit more?
Yeah, I of course. I have the one killer question. So when will it work?
I mean, investment is kind of a key question there, you know, and there's a lot of steps that you have to go through to demonstrate that something works, and there's always no guarantees at the beginning, right? I think the results are very promising. We've, you know, been able to, you know, convince people that this is something worth studying further. So we now are able to try and do the next step, which is like a semi-field release to see whether it will work in a gigantic cage. It works kind of in a small cage now. I could say that you can get it to spread through, you know, something this size, but now we're talking about something like 10 times the size of this room. If that works, then we can try to do a field trial. And if that works, then we have an intervention. So the process is slower than one would have liked. It always goes faster if more people are working on it and there's more funding. And I think we should, I hope that the concept is also something which people get excited about, because there are lots of other microbes out there that could have similar properties that we need to be investigating.
That sounds like 5 years from now. And there's another mean question. Wolbachia in dengue vectors does work. So they did clinical trials on that and they get some, I don't know, 70, 80, 90% reduction. the concept of symbionts being there in Anopheles vectors, that's quite old. But it never has been reached reality as far as I know, or it never has reached implementation. And you could also summarize the transgenic mosquitoes, the sterile mosquitoes. So I said it's a mean question.
Yeah.
What would make your approach more promising?
Okay, the Wolbachia approach, let's start with that, is been very successful in Aedes mosquitoes. Obviously, a completely different group of mosquitoes. They are naturally, many of them are infected with Wolbachia, and it survives very well in Aedes mosquitoes. Anopheles don't have much Wolbachia. Very few species have them. Anopheles, the ones that are major vectors, don't seem to have them. Wolbachia is also kind of more of a virus protection agent. So that has not been widely successful in Anopheles, despite a fair amount of interest in trying to do something with Wolbachia in Anopheles mosquitoes. And we probably have focused too much on Wolbachia because everyone got so excited about it with dengue, when there are lots of other very interesting symbionts that are in Anopheles mosquitoes that we didn't know about from some other system. So that's where I think doing something different, it may have a greater chance of some success. Other strategies, I mean, I think that these things have been studied for a number of years, but not with a very high intensity. And I think that in terms of research on the whole, if you look at the whole cycle of malaria, where's most of the research been? And not so much on the mosquito, and if it is on the mosquito, almost entirely on ways to kill the mosquito, not really on other sort of strategies. So I think that if we can shift that a bit, then that, I mean, I would say that we haven't had a fair opportunity to get any of these things to the stage of making them an intervention because it's not really been invested in. So that would be my response to that question.
Maybe I add some piece to your answer?
Yeah, please.
Could the, I don't know how you say it, the low or the absence of virulence, could that also be an argument of the microsporidia, that this time it might work?
I think that's really a key. So yes, basically microsporidia do cause disease in mosquitoes, and there's about 5 or 6 that have been found in Anopheles mosquitoes before, and those ones were found pre-molecular era because the mosquitoes were just dying and they were, spilling out these spores and stuff. It was very obvious. Now there are other ones. The one we found a few more as well, which went completely undetected for the last 60 years that people were kind of somewhat studying mosquito pathogens because you can't see them. You have to use either microscopy or molecular techniques. So yes, I think that's what's completely different about this versus other microsporidians. This is really a symbiont. It's something that doesn't really kill mosquitoes. And if you look at at least in insects, but I would say probably in all organisms, actually the diseases, the infections that reach a much higher level are those which are symbiotic. Most of the highly pathogenic ones, they are 1 or 2%, kill a lot of mosquitoes, but they don't really get to very high frequency because they kill the host too quickly, sort of. So.
Why would you call them symbiotic? So what is the advantage of the mosquito to have the microsporidia?
Yeah, okay, so
that would be the symbiotic. You would have to have an advantage of both organisms.
Yeah.
In order to say that, right?
The nomenclature in this field is a little bit confusing because actually the root of symbiosis just means living together. So actually, if you go to that sort of definition, even a disease-causing agent lives with its host for a while. So what I consider a symbiont is generally something that doesn't have a strong effect on fitness, and in some cases is beneficial. But a lot of things even go both ways. You can be a little bit negative under certain circumstances, and then in another circumstance you're positive. So, you know, it gets a bit confusing. In this case, with Microsporidia MB, it doesn't cause any effect on mosquito fitness in the lab, which is also potentially different from the field. It's very hard to study fitness effects in the field. In the lab, the mosquitoes actually develop a bit faster when they have Microsporidia MB, so it might be beneficial. It might be doing something beneficial. But then in the wild, you see these fluctuations, which tend to suggest maybe there is also, under certain circumstances, a negative effect. So it's, the names are confusing. Also, the subject area is one that is quite confusing, but it's a very good question and something we have to look into to figure out how this could work.
So maybe I didn't get it. Very nice talk, thanks a lot. So how do the Microsporidia kind of challenge the malaria pathogens? Is anything known about what functionally happens within the mosquitoes?
Yeah, also a good question. And the reason I didn't really talk about that is mainly because we don't really know. There are, of course, some changes in gene regulation in mosquitoes, which could, by looking at the genes which are dysregulated, you could say that might be having an effect. But it's not at all conclusive that it's that. It could be some other mechanism as well. This is all stuff which we're hoping to look into. I hope also other people are going to do a bit more research in this area, because it's very important to figure out what the mechanism is. Studying things, endosymbionts, so that live inside the cell that you can't really do the genetics on, and it's also a little bit difficult to sort of isolate them. That makes it a bit difficult. I think we can definitely do it. The caution is that Wolbachia, we don't really know how Wolbachia protects cells from viruses, but we already have done a clinical trial and we know it works. So, we may figure it out quickly, or it might take many years. That maybe doesn't have to hold back the idea of actually using it, but it's super interesting to try and get to the bottom of that. Yeah.
Last question, Mr. Eder, when I see it correctly.
How do the mosquitoes get infected by the bacteria?
How do they get infected
by the reproduction, or
Yeah, this is an excellent question. Also something that we're trying to figure out. So there's a lot of things that we don't know enough on, partly because it was kind of discovered 2 or 3 years ago, and it's a completely new system, and that's something which we should know. As far as we can see, it doesn't have a sort of environmental transmission phase, but there may be. So what we know is that mother infects offspring. So obviously females are a great carrier because they infect their offspring. Males would have then been kind of a dead end because they don't lay eggs, but the Microsporidia has been quite clever and figured out how to use males and then get back into a female through a sexual transmission route. So those two could be enough to explain why it stays in at the frequency it is. There may be another one which goes through the environment. Some spores are released by a mosquito and then taken up by another. So far, we've no evidence of that, and the sort of very preliminary experiments we've done didn't show that there was that type of route, but there could be. And it would be useful for us if there is, but we don't know that yet. Yeah.
So thanks, Jeremy, and thanks, Frank. And we're a little late, but I can tell you I will give up some part of my time afterwards. There won't be a big roundup or anything. So the time is devoted to Janis and Alex. So we're now reaching out for the stars, and we do that literally. And we're breaking the wall to mapping cancer using multispectral microscopy. And so since I was a little kid, I'm fascinated by space programs and astronomy. If you ask me, should we fly to the Mars? Of course we should. So what else should we do? So for me, I've got a soft spot for that. And as a medical person and a journalist, I spent many, many, many years following up what's happening in cancer. So now we have the combination of the two. For me, it's a gift after a long day. So please welcome Professor Janis Taube. She's the director of dermatopathology at Johns Hopkins University. And her lab focuses on characterizing the local pre- and on-treatment tumor microenvironment in pathology specimens using techniques ranging from routine histology to new multispectral tissue imaging platforms. And since a couple of years, she works together with Professor Alexander Szalay, who is the director of the Institute for Data Intensive Science at the same university. So he's a physicist and a cosmologist working on the statistical measures of the spatial distribution of galaxies and galaxy formations. So that's reaching out for the stars, I would say. The two of them partner in a project where astronomy meets tumor biology, and with a platform called AstroPath, they built two-dimensional maps of the tumor microenvironment in metastatic melanoma, identifying a signature that is highly predictive of response to immune checkpoint inhibitor therapy. See, we're going back to life, and patient outcome. So, welcome the two of you, and Professor David Kapper, he is the matching talk partner from the Charité. He's a pathologist like Janis, but he focuses on neuropathology and brain tumors as a senior consultant here at the Charité. And some part of his time goes into the development of DNA methylation-based brain tumor classification and how machine learning and artificial intelligence can help us distinguish the many different kinds of tumors. So with Janis and Alex, he shares experience in dealing with huge data sets, and he will guide us through the discussion with our two winners here at the Falling Walls and present his own work. So now open the stage for our guest from the US.
Thanks, everybody. Thanks to the organizers, Christiane. On behalf of Alex and I, we are very honored to be here today. So this is the story of our breakthrough, the AstroPath research project, and how astronomy helped accelerate pathology. So checkpoint blockade was alluded to earlier in the discussions. It was 2013 where cancer immunotherapy was recognized as the science breakthrough of the year. And that's when it really joined surgery, radiation, and chemotherapy in terms of the armamentarium against cancer. What's slightly different about immunotherapy, and specifically immune checkpoint blockade, as noted here in this quote by the Nobel Prize Committee in 2018 for Jim Allison and Hanjo, is that it really accelerates the immune response against tumor. So it's really a secondary effect. So rather than being directly tumoricidal, right, you get the immune system going, and then the immune system actually helps clear the cancer. It's incredibly exciting, and we've seen amazing responses across numerous different tumor types. However, we still need biomarkers. And why is that? It's unfortunately now, if you really do look across all tumor types, less than around 20% of patients actually respond. And it is true that they have fantastic and durable responses when they do respond. But we still need a better way to continue to develop these therapies and to match the right patient to the right regimen from the start. And so these also, because of that secondary effect of getting the immune system going before the immune system potentially attacks the cancer, often have longer delays in terms of therapeutic start and apparent efficacy on radiography. There's also sometimes a paradoxical pseudo-progression, as we would call it, where as the immune system inflames the tumor, it actually looks like the tumor is progressing before it ends up regressing. And so if any of this happened while you were on chemotherapy, for example, you would get pulled off of your regimen immediately, especially if new lesions end up appearing while you're on therapy. And yet this is a phenomenon that's recognized on immunotherapy. So if you had a biomarker that helped give your oncologist confidence to leave you on the regimen, that would certainly be useful. These also have attendant effects. So you rev up the immune system, there's immune-related adverse effects. Sometimes these are quite consequential in terms of long-term immune-mediated damages on the patient's organs. And so you certainly don't want to give these to patients unlikely to respond. One of the other cost-benefit ratios is literally the cost. And so in the United States, these regimens can cost over $100,000. So a huge expense to the healthcare system as a whole. And also for those patients who aren't responding, we really want to put together combinatorial therapies. And right now, everything is being combined effectively empirically. And so if we are able to come up with biomarkers to develop rational combinations, that's incredibly desirable as well. In terms of the biomarkers that have been put forth to date, there's PD-L1 immunohistochemistry, which our group was involved in. This is really labeling a single ligand of PD-1 and read most often as simply positive or negative beyond a given threshold. There's been gene expression profiling that's been looked at, most commonly interferon gamma gene expression. And then in terms of tumor mutational burden is the number one that has been brought forth. We've put a lot of our efforts now into multiplex immunofluorescence and immunohistochemistry. And this has really allowed us to spatially resolve multiple markers at the protein level and also on a single cell level to better characterize the complexity of the immune system as it interacts with tumor. And this really forms our guiding principles. Again, having that single cell resolution allows you to look at complex co-expression profiles. It also allows you to understand the spatial biology. And so the proximity of different molecules to each other, different immune cells to each other, as well as to the tumor itself. And we really believe that the next generation of the tissue-based biomarkers are likely to be identified using well and curated data sets, perhaps with these multiplexing technologies. And so we were very excited as these technologies started to emerge, especially ones that allowed us to work very readily with formalin-fixed and paraffin-embedded tissue. We didn't need fresh tissue anymore, and this is really what surgical pathologists work with. We have archives that are well annotated going back decades, allow us to do like quick follow-up studies on the implications, both in terms of prognosis and predictive value of any identified biomarkers. And yet, when I first started studying these technologies, I am a diagnostic pathologist. We're used to reading the entire slide. And so these technologies allowed you to look at single high-powered fields. And so typically people looked at 5 or 10 high-powered fields, including us in our earliest studies. And what we want to do, and what, for example, the typical immunohistochemistry allows you to do is look across an entire slide. And the cost was exorbitant, both in terms of person time as well as computing capacity. So this is a single lung cancer specimen, and one tumor took over 1,000 of these high-powered fields and 300 gigabytes of disk space. So it would take me 20 minutes alone just to move one of these high-powered fields from the microscope where it was acquired onto the server. So forget about doing some sort of large cohort and meaningful analysis. And it is at that point that I am most grateful that some of our colleagues at Johns Hopkins introduced me to Alex Szalay, and I'll let him take over from here for a bit in terms of this story.
Thank you. So the astronomy story started 30 years ago. So when Johns Hopkins got into a big sky survey project, the Sloan Digital Sky Survey, and it was on parallel with the Human Genome Project. Also, our first data release came out exactly 20 years ago, so again on par with the Human Genome Project. So the idea was to build a special telescope in New Mexico, cover the whole focal plane with CCD chips, which became just about affordable at the time, and then map the whole sky that was visible from North America and make all the data almost immediately publicly available and open. But also not just the raw data, but we created actually a shrink map, a shrink-wrapped version, so carefully calibrated, annotated, and organized into a database. So the idea was to build everything in 4 years, and then run the survey in 4 years and be done by the year 2000. Of course, as it turned out, scaling out on that scale, on an unprecedented scale, was much, much harder than we ever thought. So actually, the telescope started operating in 2001. It went on, the proper sky survey went on until 2008, and we are still operating, but doing slightly more extended projects. But the whole point was that the data is entirely public, and we had annual data releases, one following the other. And every institution who participated in the project, so Hopkins was one of the few universities, Princeton, Chicago, UW were the others. We each had to build certain components of the system. And since I was working on the spatial statistics of the galaxies, I said, okay, I can't do this unless the data is properly organized. So I signed up for the database, but little did I know what I'm getting into it. But essentially, the main point in the project was that we made a transition from individual astronomers pointing a telescope to a part of the sky and studying that little field for years and years, to take a big swath and essentially go from the manufacturing of scientific data into the industrial mass production. And that's kind of so I had the privilege to bump into Jim Gray, who, as a Turing Prize-winning computer scientist, he invented database transaction processing. Every ATM machine in the world works with his algorithms. And then he spent 50% of his time over the next 10 years working with me. And we built an experimental system. How can we make all this data not just accessible, but not just available, but accessible? And so we created the Sky Server, which is still online today. And we had today, at this point, about 4 billion web hits. And we enabled people to a backdoor into the data. So not just the usual database interfaces, they have drop-down menus and so on, but we allowed people to write free-form SQL queries. You can run basically any SQL query that you want. People said, you are crazy. No astronomer will want to do this. In 3 months, people were basically writing beautiful stuff. And so people wrote 500 million SQL query over the last 20 years. We published 9,000 refereed papers and about 500,000 citations. And there are only about 50,000 professional astronomers in the world. But we had 7 million users. Actually, I think the most up-to-date number is closer to 10 million. So we saw the emergence of the internet scientists. So a Dutch school teacher made on the Sloan data a unique discovery, and she was part of all the papers appearing in Nature and Science. A group of computer gamers discovered a new class of galaxies that everybody missed. So it really showed that transition in science. And it is still, the system is still the world's most used astronomy facility. So AstroPath. So we met with Janis, and it turned out that, just the same way as galaxies and stars are the atoms of the universe. So they are kind of the elementary particles that everything is built up from. The cells are essentially the building blocks for the tumor and cancer immunotherapy. So in many ways, it turns out that a lot of the questions Janis wanted to ask from the data were quite similar in terms of the spatial relationship. So where are the different types of cell located? What is close to what? And so it was studying the tumor microenvironment was actually not that far off, surprisingly, from the study of galaxies and stars. And so here we tried then to what does it take to actually do the same leap into the Industrial Revolution, to do a non-incremental study. So this is really breaking down two walls. On one is, taking probably two disciplines of science which are probably as far as apart as you can think, astronomy and cancer research, and then also the breaking the wall of kind of the incremental evolution of the techniques and technologies. So essentially our goal is to increase the data collection by a factor of 1,000, so not by 2 or 4 or 8. And essentially, this really parallels the sky surveys. So I see the task is that kind of provide a disruptive assistance. So disrupt but assist. And so let's figure out a way together how can we actually apply the same techniques and scale it out the same way as we learned. It took us 20 years to figure out. And so let's not reinvent the wheel, but let's learn from the mistakes we made. And one of the keys to have an interactive way to visually browse the data, like Google Maps and MapQuest was. So to create the same thing for not just for the sky, but actually created for the tissues. But what is the spatial information? It is partly, the location of the cells, but we try to do one better. We are storing actually the outline of every nucleus in the data and the membrane of every cell. Today we have 500 million cells in the database. And the Sky survey cost about $240 million to date. We process through 6.5 trillion pixels. We are now at around 5 clinical cohorts that are in our database. We have to now process through more than 10 trillion pixels, actually, so twice as much almost as with the whole Sky survey. And we represent all sorts of fancy spatial information, and we had to automate. So we had to eliminate as much of the human labor that goes into, because people make mistakes and make the data inconsistent. This was, in a sense, the hardest. And we have lots of databases, and it is linked to the second generation Sky surveyor, which we call now Sci surveyor, where you can actually access the data from programmatic environment, from Jupyter, IPython. You can run AI tasks, you can run Keras and TensorFlow, or you can access the data from R. So this is kind of where we are today. And I would like to hand back to Janis, where she now talks about the results. How did we use these technologies?
Okay, so I talked about some of the merits of these technologies. When they were first described to me, they were described as flow cytometry on tissue. And the output of flow cytometry really is fluorescent intensity in most instances. And in fact, that's not how these were being used. They were being read as simply positive or negative for any given marker. And so part of what Alex did that he didn't even talk about was all of the optical corrections and standardization that allowed us to come up with a very robust measurement of intensity in situ across an entire slide. And so there was mention of the 2D maps that we made before. Here's the 2D map, but we were able to also do visualizations of marker intensity in 3D. This is important in immunotherapy and immunology in general. Marker intensity shows different functional states of the cell. So PD-1, which is the direct target of many of these checkpoint agents, when it is expressed at low levels, as you can see here, this is the advancing edge of the tumor, is when the T cells are starting to get activated. And then as they move further into the tumor, they see chronic antigen exposure and the like. They're becoming so-called exhausted, and they express PD-1 at very high levels. And those are not able to be recovered by therapy. We think it's these earlier ones that are affected by the monoclonal antibody therapies. So very important biomarker implications to be able to parse those different levels of expression. Here's an example of one of the tumor and immune maps that Alex helped us generate. This is a melanoma deposited in the lymph node, and we were able very much to identify this front here where the immune system is interacting with the tumor, and you can appreciate the very organized fashion in which it's doing so. We were specifically able to map these different populations as they related to the tumor and stromal border. One of the most interesting things that we found was this population of CD8 FOXP3 positive cells. And so in the literature, there had been some reference to these, and the hypothesis was that these were actually suppressors of the immune system. There's a very well-recognized CD4 positive, FOXP3 positive cell that's suppressive, and the hypothesis had been that this was just a CD8 variant of that. Well, in fact, it was localized immediately to the tumor stromal border. And based on some of the localization and other features that we found, we hypothesized that it in fact was an early effector T cell. And we've since gone on to confirm that with single-cell RNA-seq studies. But it was really the accurate labeling and mapping of these that led to that new discovery of functionality of that cell population. So that cell population, as well as a couple of other defined features, really allowed us to identify these three different phenotypes of the tumor microenvironment, actually using only 6 markers. And so we were able to identify those that were associated with the poor prognosis for patients with melanoma receiving immunotherapy, those with good prognosis, and then the remainder of those that had an intermediate prognosis based on the way that the immune cells arranged themselves and interacted with the tumor. And these are the results of those. So the receiver operating characteristic curve, the area under the curve that we're aiming for, perfect, is 1. By combining those features that I described, we were able to get a remarkable AUC of 0.9. And of the different tumor microenvironment phenotypes that I described, for those who aren't used to looking at these are Kaplan-Meier plots. And so this is essentially a plot of patient survival after the treatment is initiated. So all of these patients had advanced melanoma, and this is the proportion of patients this is their overall survival years after treatment initiation. So the blue line are those with the highest proportion of those FOXP3 positive, CD8 positive cells. Those patients did extremely well after 5 years of receiving a PD-1 regimen. We have those patients with the worst prognosis, you can see obviously here, did not do well at all, and those with the intermediate prognosis. And so this worked for 2 different metrics of long-term outcome. This is overall survival and progression-free survival. This was in a Hopkins cohort, and we then validated this biomarker in a separate, completely independent cohort from Yale University to similar effect, a very nice high area under the curve, as well as stratification of the patients in terms of long-term outcome. So that was our first cohort. As Alex mentioned, we now have a number of different cohorts in the database. One that we're particularly excited about includes on-treatment specimens, not pre-treatment, but those after treatment, which really gives us insight into how these immunotherapeutic regimens are exerting their effect on the tumor microenvironment. And so this is from a patient with lung cancer. The blue line represents where the tumor used to be. The green line represents where the residual tumor still is. So you can see we've got about 60% tumor clearance. And we've been able to map different populations of cells in terms of the T cells interacting with the residual tumor. But one of the most remarkable features that we can see are really the characteristics of tissue repair. This is wound healing between where the tumor used to be and where it still is. It's the body's trying to repair itself now that it's cleared the cancer, which is quite remarkable. And so we're able now to understand that better and perhaps recreate that tumor microenvironment to help in certain instances where the body might need a little push to help clear the cancer. So I've been incredibly lucky and benefited by Alex's expertise and what they've done in terms of the Sloan Digital Sky Survey, moving from the telescope, whole sky, cosmic web, galaxies, and spectra, over to now being able to map whole tissue of tumor by cells, and as he noted, even subcellular components. We're looking at adding in genomics as well as spatial transcriptomics now into the database. This is really a unique facility, and by the way that we're tiling it, we not only have the 500 million cells that Alex alluded to, but also over 336,000 of the individual high-powered field titles, and that's really allowed us to ask very important questions about sampling tumors for diagnostic effects, which has been a big outstanding question in the field. And so I'm incredibly excited to have the opportunity to present this to you guys today. Thank you for your attention. I'd like to also acknowledge all of the wonderful team members on both of our sides, as well as some of our pharma and commercial partners, and of course our funding agencies who really got excited about this concept of astronomy and pathology.
Thank you so much. So I hand over to David Kapper now.
Thank you very much for this beautiful data and also this very nice to follow talk. I very much enjoyed that. And I'm one of the poor pathologists sitting at my microscope doing PD-L1 immunohistochemistry almost every day. And now knowing what is out there, and I haven't even heard about the FOXP3. Well, I saw it in your paper, but this is of course, it's a step from before what we're doing, but I'd like to discuss with you later on how this could be integrated into our daily work also, maybe across the Atlantic here. What I will do is a very brief demonstration of what we are doing. I will show a few of the parallels that we're actually doing in our mostly Germany-centered pathology work. I will shorten this up because for time reasons quite substantially, so please mind with me that we will rush through these slides a bit and then come to the actual discussion again of your data, just because we're a bit late. There's a disclosure. You can ask me about that later on if there's interest. One of the big parallels that we are doing and that I personally am working on is that we're trying to standardize. This is histopathological images, and what you just briefly mentioned here is one of the big works of Alex was actually to define cutoffs for various things, because pathology is a quite subjective field of medicine. A lot of therapies are then based on these subjective measurements, and I think it's a big task at the moment to really try to come to an objective way. This is different brain tumors that I'm focusing on in my work. There's about 90 different of these, and the training of a pathologist to a large part is actually to identify what name belongs to what of these tumors, what treatment should these patients actually get. And of course there is a substantial subjectivity, meaning if you get your cancer diagnosed in France, it will be different to Germany, etc. And even within Germany, depending on what center you get your cancer diagnosed, you will have the chance of a different diagnosis, potentially with very different treatments that you will get administered. And that's a big problem that we try to solve using DNA methylation, which is a central marker of cellular differentiation. As I said again, I will go across this very, very briefly. And what we could see in the earlier studies is that this DNA methylation mark is actually something that gives a code to cellular identity, and that we could use to separate out different cells of the human body. If we had a big atlas, that we could then do DNA methylation profiling of single cells and tell you this is a brain cell, that is a gut cell, but we can also use it to differentiate different types of cancer. So this started here with brain cancer. We're currently mostly working on other types of cancer. My main projects are, for example, on breast carcinoma and lymphoma at the moment. So this is further expanding. What you use, and that's a second parallel thing, and this is also why DNA methylation has entered the stage for tumor classification, is that you can use paraffin-embedded and formalin-fixed tissue. This opens up all doors to the archives, and it is a relatively cost-inexpensive technique with about $400 per sample. Of course, that's different to other things that you've heard before. And so the general concept of what we've been doing is we try to translate everything we know about brain tumors into a huge cohort of DNA methylation profiles. So we profiled back then over 10,000 human brain tumor samples and put them all into the central database, then did some unsupervised identification here of distinct subgroups and could realize that they're very much in parallel to what we've seen in histopathology. So they're color-coded here by the histopathological diagnosis. And for the first time got a map across the different brain tumor classes that we can separate. And this done entirely without histopathology in this case, but done entirely on these molecular data. This has been diagnostically implemented and using machine learning. And again, I will not go into detail here. You can do nice testing of such an algorithm and see that it has an internal misclassification rate of maybe 1%. And you can also then compare this to what would it look like if you compared it to the pathology workup of samples. And we did a prospective trial of over 1,000 brain tumors in this case. And to also cut this short here, the system will lead to a reclassification of 12% of brain tumor samples, meaning that if you cut it very short, the pathologist, and that was in this case me and the team that was done in Heidelberg while I was working still there, that in 12% of cases, in fact, we really changed the diagnosis of the tumors that we came across. Another 12% of cases, such a machine learning tool told us it doesn't recognize this tumor, and these are the ones we're currently focusing on for the discovery of new brain tumor entities. One of the other central things is that the data is very easily shareable. So the internet was actually the big motor behind this thing. A website was put up where people can upload their locally generated DNA methylation data. This is hosted by the German Cancer Research Center and University Hospital Heidelberg. And within a few minutes, you get an automatically generated report using the machine learning and also additional molecular data on this. And this has been used just under 100,000 times to date for the diagnostic classification of such a sample. The website is for free, but the nice thing about these data is that you can share the data while uploading, so you don't have to, but you can cross this, meaning that there have now been about 90,000 tumor profiles that have come together in a central database and allows now the drawing of very, very much more exact maps of brain tumors, which has now led to the discovery of over 30 new brain tumor classes, and several of these have now officially also been integrated into the upcoming WHO classification for brain tumors. And this is also one of the things that I started at the Charité in 2017. One of my tasks was actually to introduce this technique also here at the Charité, and we've done so. We're currently doing about 50 to 90 diagnostic cases using this technique per month. And as I said before, brain tumors is one field, but this is very rapidly expanding into other fields. This is an example here of a sarcoma classifying tool, and currently we're, as I said before, mostly working actually on a large carcinoma classifier. So at some stage we believe there will be a giant map of all types of epigenetic kind of codes for the different types of human cancers. And again, the data is very easily shareable. All the data that was published in the initial presentation are publicly available, and so a lot of people are playing around with this at the moment. With that, I would like to thank the people here mostly involved in this project here, and also you for your attention so far. And with that, I think we would go back to the discussion, if that's okay. So I didn't have a time, so I actually have no idea how long this took. But maybe the two of you could enter the stage again, if that's okay.
Yeah. Thanks for doing this, David, because I think it's bad to actually ask you to cut down a little bit, but I think
I hope you survived of that.
Okay, you start.
So maybe I could start. So I'm super fascinated and super jealous about this massive database. So I would like to, of course, hear a bit more about how that is structured and organized. But maybe you could could tell us actually really a bit about how this is done in a practical term. So you generate the data. Is this, I mean, how is this translated over? Do you do this really on a daily basis? And maybe just to give us a bit of insight on.
So it starts on your side. So.
Well, so it does start on my side, but it was overhauled by Alex very early on. So, you know, rather than spending $70,000, $80,000 on a postdoc one of our first years together, instead we upgraded to these 10 gigahertz switches to help move data. And we were able to hook into what's known at Hopkins as the Hornet, which then transfers data at very high speed to where Alex is a couple of miles down the road at the undergraduate. And so from there, he took care of all of the really impressive data organization, and I'll let you take over.
Yeah, but so the most important step was to essentially formalize, so turn all the patterns that we learned in astronomy into processes. So that how to develop the protocol, how the data should be processed as it leaves the microscope, what corrections do we apply to the images, and then how do we convert it into how do we transform the data into a database loadable format. And then I had to create a database schema, then write an automated loader, which consisted of probably 20,000 lines. Mm-hmm. So, but most of the code was actually error checking. And we developed that's another notion we learned in astronomy, a so-called two-phase load. So we treated every slide first as a separate entity. We loaded each slide into its own little database, and then we basically run all the checking and testing code. And once it was validated, then we actually migrated into the data warehouse so that we don't have to unload it afterwards. And then we had to create the images, so we had to do our own image stitching of the mosaics, and then out of this we created something called a deep zoom hierarchy. So we basically broke it up into an image pyramid of tasks, loaded that into the database again, and then we had to develop the front end, the visualization front end. So
if you had now today had a diagnostic sample, how long would this process take? Has this been fully automated or?
Almost automatic. We still have some intervention needed, human intervention at the segmentation, but we are working very hard to replace this with a neural net, so deep learning. So,
really great work. Maybe I would go to the audience and.
For actual therapy or diagnosis of the patient, yeah. So how did you get, for example, the image that you showed in your last slide? Was it in a living patient with this high resolution, and how is it done to detect the fluorescence?
Yes, so there is a multispectral microscope that we use, and there are a number of different platforms that are out there now for multiplex immunofluorescence that are able to acquire images at different emission wavelengths for the floors, and that's the goal of how they're able to help and separate those.
This cannot be done in a living person, right?
No, not yet, unfortunately. Although they certainly are looking at radiological ways to label patients. But no, these are from tumors that have been removed from patients and sent to pathology, and they have chemical processes performed on them. And then they're made into slices that we're able to stain with different markers and fluors.
Okay, but in these days, surgery is not a major way to go. So this is an exception in these days, right?
Well, for patients with advanced disease, metastatic disease, you often do have at least a biopsy to confirm what type of tumor you have. And so those are the ones that we are then looking at specifically in the study for melanoma. What I showed you at the end with lung, since we've had these wonderful successes with patients in the advanced disease setting, what they're now doing is moving these therapies earlier and earlier. So now, in theory, if a patient comes in, for example, with a newly diagnosed lung cancer, they may give 2 cycles of immunotherapy. The idea is that you potentially shrink the tumor, but you're also able to prime the immune system while the tumor's still in place. And then you go on to have the definitive surgery. And so for us, we then get that tumor that has now been exposed to immunotherapy for 6 or 8 weeks and are able then to do extensive studies on how the therapeutic mechanism of how these therapies work. And so that's how we're getting a lot of our specimens there. You can then send for genomics and transcriptomics as well as some of the protein mapping that we're doing.
Hi, thanks a lot for all the great presentations. 2 quick questions. The first one is, do you rely on a commercial system for your immunofluorescence staining? Is that the Codex or whatever platform, or do you also develop your own immunoreagents and to stain cells?
Yeah, so we did use commercially available materials, but what we found was that when we used the reagents as the manufacturer recommended, we were not able to achieve staining that was equivalent to the chromogenic stain that I use in my CLIA certified lab every day. And that was really important to me as a diagnostic pathologist. For example, we alluded to PDL1. If somebody's going to say that they do PDL1 on multiplex along with 8 other markers, I think the default understanding is that the PDL1 is going to be equivalent to if you did a single stain, and in fact that wasn't the case. And so what we did was we tested a number of other secondary antibodies and actually changed those out to be able to make sure that we had that equivalent. So our reagents are commercially available but not used as recommended. Took some mismatching to get it the way that we thought it needed to be standardized.
And the follow-up would perhaps be naive because I'm not a pathologist, but you mostly target or stain for the targets of your therapy, or does your staining and the whole huge amount of data you generate also have the potential to reveal new potential targets or unexpected biomarkers?
Yeah, so the hope would be unexpected biomarkers, of course, in the end, and certainly we're using a lot of the single cell techniques to potentially reveal that. At some point, we do prefer monoclonal antibodies to run in our assays to validate any targets on that. And if, for example, something that came up didn't have an antibody available, we'd look for something else in the pathway or the receptor or some other way to label it and be flexible in that manner. But we're definitely using either antibodies that are already in existence or informed by other methodologies.
One last question.
One last question.
Yeah, thanks for the wonderful talks. And while we have you both on stage here, epigenetics and transcriptomics and single cell, right? I mean, the next step is logical. Yeah. Now you have the immuno, and you probably integrate, and probably you come together. So that is maybe planned or not yet?
We'll try. Yes, but I think that's a great closing remark. In fact, so I'd love, as you may know, there's a large tumor family called astrocytoma in the human brain that I'm actually focusing on a lot. So maybe if you would like to add an astropath project to your list, I would be very welcome to join into that. I think our time is up. So with that, I would like to close the session and hand over maybe to, do you have any last room, Christine?
Only a very few. Okay. So thanks for that.
Thank you for your patience.